สวัสดีค่ะ Good morning and good afternoon from some area of the attendees. Ladies and gentlemen, welcome to the webinar Small Things but Giant Steps to Bio-Circular Green Economy and Resilient Economy. So with me, my name is Rungpet Chitanuwat. I'm going to be the moderator for today's session. So we have a lot of well-known speaker and expert in the how to do the food sector in the sustainability way. On behalf of Thailand Institute of Scientific and Technological Research, or TISTER, I would like to thank to all organizations to support this webinar, including Bioresource Collection Research Center, BCRC from Taiwan, Food Industry Research Development Institute, FIRDI from Taiwan, Department of Bioscience, Horst University from Denmark, Green and Company from the Netherlands, Leichhardt Technological Center from Spain, and last but not least, another two organizations that make this event happen: National Research Council of Thailand and World Association of Industrial and Technological Research Organization, who is the global network of research and technology organization that promotes collaboration among its members towards the SDG. So ladies and gentlemen, you are with me here today till 4:30 in the afternoon of Thailand time. I will need to apologize if I have to communicate in one time zone only. So my timing of my moderating session today will be based on Bangkok timing, GMT +7. Today's webinar will consist on the two parts, and the start of welcoming remark and agenda will be the session 1 on microorganism and utilization of food industry, microorganisms and their potential application trends. Then will be on the research and application of microorganism of food, Bioresource Center, and underpinning the bioeconomy development from Taiwan. Probiotics 101, Precious Life Inside Us, and Technology Transfer of Microorganism Research Case Study. Then we will have the session 2 that is about insects as a trend of alternative protein source. There will be a video from our Scarrat Environmental Research Station, or we call SERS, which is under TISTER. Then followed by the global trend, edible insect as an alternative protein of future food source, linkage to SDG and resilient economy. Then going to have the speaker from the Netherlands talking about business opportunity with ant farming. And we're gonna have a special session that have the White Throat Innovation Award winner to share with us on the smart in food, insect-based food source to supplement nutrient deficiency in vulnerable area. Before we get started, the housekeeping rules today. If you have any question, please put in the Q&A box. If you could not find the Q&A box, you can put in the chat. Our administrative team will bring the question to the session. And then I would like to inform you, must be looking for the handout. You can go to the chat box. We provide a QR code that you can download the handout of today's webinar. And I would like to ask everyone, if you stay with us for the whole day, please make sure you have a glass of water. Make sure you're stretching your body during the webinar. Your well-being is very important for the host of this session. All right, let's start the time for the webinar today from our governor from TISTER on promoting the bio-based economy. Please have a look on the video and we come to the organization.
Thailand is a country containing incredible biological diversity. However, our natural resources have been continuously depleted over time. Sustainable practices to protect natural resources while utilizing conservation is our focus at the Thailand Institute of Scientific and Technological Research, TISTR. TISTR is a state enterprise under the Ministry of Higher Education, Science, Research and Innovation, or MESI, and realizes the importance of biological diversity protection. It implements both in situ and ex situ conservation. Focusing on in situ conservation, the Sakerat Environmental Research Station, SERS, established in 1967 and located in Nakhon Ratchasima, northeastern Thailand, covers an area of 78.08 square kilometers of tropical forest. Designated as the Sakerat Biosphere Reserve under the UNESCO Man and Biosphere Program, SERS is the first biosphere reserve in Thailand and one of many among Asia and Pacific countries. Since its establishment, the station has intensively protected biodiversity. SERS has continuously and effectively protected forest areas as well and maintains a high level of flora and fauna biodiversity. The areas contain over 1,000 vascular plant species, 400 invertebrate species, 500 species of vertebrates, and includes more than 250 species of birds. Conveniently, SERS is one of the most popular places to observe the incredible Siamese fireback, Thailand's national bird. Moreover, this area is a locality for over 50 unique species, drawing many researchers from both Thailand and abroad to conduct projects focusing on the ecology of tropical forests and animals. There have been more than 800 articles published both domestically and internationally on the Sakerat area, making it an important conservation area for learning, research, and ecotourism for people all over the world. In addition, the station has a mission to promote and exchange knowledge on the sustainable use of local natural resources. SERS supports the cultivation of economically viable trees in combination with indigenous mycorrhizal fungi to help accelerate growth rate and reduce tree mortality. This also increases the yield of edible mushrooms in the area. The station teaches local communities to cultivate Melanthea suavis, a high-value vegetable, and other edible wild plants. SERS also conducts training on weaver ant farming to increase the income for local people while creating jobs and careers. This helps establish a basic and crucial understanding of the natural resources available to the communities, which is fundamental for people to live sustainably. In terms of ex situ conservation, TISTR has established the Community Seed Bank at Lam Takong Research Station, LTRS, in Nakhon Ratchasima. The seed bank collects more than 200 agricultural, indigenous, and wild plant species. There are 20 seed types under conservation that can be only found within Thailand. Including various herbs, household vegetables, and fruit crops. Over 300 seed types have been planted at the first and second Chalerm Prakiat greenhouse buildings of LTRS. At LTRS, we also study insect biodiversity. This includes breeding rare insect species, research on insects as a food source, and the utilization of insects for agricultural purposes. The Tropical Insect Sanctuary at LTRS is a learning space focusing on various species for researchers, students, and interested people. In addition, TISTR has a culture collection center, also known as Bangkok Mersen, and is the first collection center in Thailand of its kind. It was established with the support of UNESCO to strictly store and conserve various microbial strains found within Thailand and other countries. Over 11,000 strains are collected in its database and are available for various purposes. The Culture Collection Center provides testing and analysis, deposit of microbial strains, intellectual property application in terms of entrepreneurial research, studies on the utilization of microorganisms, identification and characterization of novel species, and selection of potential microorganisms for bio-based product development used in the agricultural, medical, food, energy, and environmental sectors. TISTR's dedication to promote both in situ and ex situ conservation, focus on research and development, and building infrastructures to leverage a bio-based economy is in line with the BCG economic model of the country. TISTR commits to the natural protection and conservation of biological diversity for the next generation.
All right, that's it about the TISTR, or I always call TISTR. So the host of the event today, TISTR video presentation that is really show the dedication to promote bio-based economy according to the country policy bio, circular, and green economy. So it's time now to the first session on small things but giant steps to biocircular, clean BCG and resilient economy, food and wellness for the future and business opportunity webinar hosted by TISTR. I would like to invite Dr. Shrutima Iamchochawalit. She is the governor of TISTR, Thailand Institute of Scientific and Technological Research, to deliver welcoming remarks. Please, Dr. Shrutima.
Distinguished speaker, participants, ladies and gentlemen, good morning. On behalf of TISTR, I would like to warmly welcome you all to this event. Over decades, people witnessed the greater competition from limited natural resources and look for the safe and secure food supply. Whereas climate change is concerned as primary impact to the production system of agriculture, forestry, fishery, and aquaculture. Moreover, it has been almost 3 years that the world has changed drastically regarding to the COVID-19 pandemic. The scale of impact is ranged widely, covering the terms of economic, social, and environment. This is why international forum and high-level dialogue platform have intensely raised up actions to build resilient economy, clearly be seen in the UN SCAP, where addressed that the policymaker need to think of strategy to improve resilient economy, as it is ability to help country to absorb shocks, adaptation, mitigation to change, and prevent systemic breakdown. Instead of turning back, building forward toward more resilient, inclusive, and sustainable economy must be done. Asia and the Pacific is the region exceptionally rich of biodiversity. Unfortunately, the list is declining. Decreasing natural capacity is affecting to people well-being undoubtedly. So, the government of Thailand has officially launched Bio-Circular Green as a national agenda to achieving sustainability of the country. Thailand Institute of Scientific and Technological Research, or TISTR, a state enterprise and research organization under the Ministry of Higher Education, Science, Research and Innovation, has adopted and shaped our work implementations to align by its principle. We will aware crucial role of biodiversity as precious national resource and contribute hard effort on both in situ and ex situ conservation and optimal utilization. I wish this webinar is a special forum to discuss and emphasize the power of small livings, focus on microorganisms and insects for utilization in food industry and novel protein source. These are the global trend and competitive bioresources able to leverage properties of food, healthy products, including byproducts for value creation. Lastly, I wish this webinar will inspire awareness of people on biodiversity and environmental issue that our planet is facing. I sincerely thankful to all speakers on your coming and strongly encourage attendees to actively share or ask solutions from our invited experts. Let us work together as a part to build up inclusive growth and resilient economy, not for anyone, but for us all. Thank you.
ขอบพระคุณค่ะ Thank you very much Dr. Shutima for the remarks. Definitely we have to do for our future generation. So with me now we have our co-host of this event who really support to make this webinar happen. I would like to invite Dr. Bing Hill Barry Young. He is the Deputy Director General from Food Industry Research and Development Institute or FIRDI from Taiwan to deliver the welcoming remarks. So I have a bit of Dr. Barry. He received both his Bachelor of Science, Master of Science degrees from the Department of Food Science and National Taiwan Ocean University of Taiwan. He then pursued his PhD degree with food engineering emphasis at the Department of Food Science in North Carolina State University at Raleigh, North Carolina, USA. In June 1991, Dr. Yang joined Food Industry Research and Development Institute, FIRDI from Taiwan. And in recent years, Dr. Yang has led his team emphasis on process development for nutraceutical and nutrition products. In 2021, Dr. Yang has been promoted to be the Deputy Director General of FIRDI. Please welcome Dr. Yang with Dr. Barry with me for the welcoming remark. Thank you.
Thank you. Good morning, ladies and gentlemen. This is Barry Yang on behalf of FIRDI, the Food Industry Research and Development Institute in Taiwan. It is our pleasure to co-organize the morning session on microorganisms and utilization for food industry. Our institute was established about 55 years. This year is our 55th anniversary, and the institute was founded for the food and the biotech industry in Taiwan. We joined the Waitrose family a few years ago, and for years, Tista is one of our major research partners in Thailand. Bioresource Collection and Research Center, we call BCRC, is one of FIRDI's division. It was established for bioeconomic development in Taiwan. We are great to have my colleagues from BCRC to share experience in the webinar. I hope this webinar proceeds successfully and all participants have good health, especially during this COVID-19 pandemic period. Looking forward to further connection with you all. Thank you.
Thank you, Dr. Barry. Please stay with us. So it's very nice to know that your organization is 55 years. Tista also 59 years. We are really brother and sister. So thank you, Dr. Yang, for the Dr. Barry for the welcoming remark. And now I would like to invite all speakers to have a great session on having the group photo together. Please open your video. I would like to invite all speakers for today to open the video for having the group photo session. All right, I just count 1, 2, 3. Another one, please. 1, 2, 3. Thank you very much. Thank you, Dr. Barry. So right now we are going to start our first session, which is going to be from our speaker from TISTER. The topic will be on microorganism and utilization for food industry. I would like now to introduce Dr. Pandit Phangsin. He is the director of the Expert Center of Innovative Health Food, or we know as InnoFood from TISTER, Thailand. I have a little bit of background of Dr. Pandit. He has academic background in applied biology and chemistry from Tokyo University of Agriculture, Japan. And his area of expertise, I may take another one day to explain it, but I will have a short brief about him on fermentation technology, industrial microbiology, enzyme technology and applications, food microbiology, microbiological analysis in food and beverage, genetic engineering technology, bioprocess operation, probiotics and prebiotics, taxonomy of yeast by using molecular tools, culture collection management. So right now we are ready to hear from the expert, Dr. Pandit Phangsin, on microorganism and utilization of food industry. Please, Dr. Pandit.
Thank you very much. Good morning, everyone. Today, I'm really glad to have the opportunity to talk on the topic of microorganisms and their potential application 10. So just like the topic of the seminar, the microorganism is really small, but it's big and really impact promotion and contribute to the human and the world. Next slide, please. As you know, as the microorganism that cannot be seen by our naked eye, we have to use the microscope. We found them in everywhere, in the soil, in the air, in the water, in the forest, in the ocean, in the city, and even surround us and on our skin, and sometimes it's in our body. So it's really important and really affect to every aspect to the human and to the world. Next slide, please. The discovery of microorganism. The microorganism was found when the magnifying glass microscope was constructed by the Dutch scientist Anthony van Leeuwenhoek in the 1676, and he observed the peppercorn water and spices. He found the small organism is bacteria. So it's the first time of the bacteria was discovered by the man. So later on, he was recognized as the father of microbiology, and he is the first of microbiologists. Next slide, please. We can classify the microorganism into the 6 groups based on the structure of the cells of microorganism. The first group is the bacteria. The bacteria is really well known, and we can know and we can hear often. The bacteria is the prokaryote that the lack of the nucleus membrane and have the various kind of morphology, just like in the rod, cocci, sphericals, and mostly distribute in the natural environment. The second group is the fungi, that's consisting of the yeast and mold and fungi, and also is the mushroom. The third group is the algae. The higher the evolution of the group, they have the pigment of the chlorophyll, and they have the photosynthesis for their utilization for their clothes. The fourth group is the archaea. The archaea is just like the bacteria. It's the same group of prokaryote, but they're different in the cell structure. So make them is so rigid. They can survive in the extreme environment, just like high temperature, in hot spring water, in high salt, high pressure. So sometimes we call them the extremophile. The fifth group is the protozoa. Protozoa is just like the one-cell animals. We can find in the water several, sometimes just as the amoeba or mycelium. And the last group is the sixth group is virus. The virus now maybe you really well known about right now. So it is consists of the piece of nucleic acid inside the body, but they cannot survive by themselves. They need to the host cell to stay, and they infect and propagation of their genetic material for their life. Next slide, please. The total estimate of the microorganism in the world is have reported nearly about 2 million species on the world, but still really few of that can be discovered. It's only the 157,000 species have been found, just only the 9 to 10%. The big group of these microorganisms is the group of the fungi, yeast, mold, and the lichen, the big group one. Next slide, please. When we looking for the application of microorganism based on the purpose for using, we can classify the application of microorganism into 6 applications, such as the agriculture, food production, energy, industry, environment, and human health. Next slide, please. As the first application in agriculture, several of microorganisms, they have played important role to contribute to the plant growth or to support in the growth of the plant, just like the biofertilizers, nitrogen fixation, biocontrol to control of pesticides, insect, or the control of the plant pathogen. And also they have ability to solubilize of the phosphate in the complex form in the soil to be the free form to release to soluble in the soil that can plant can be used support for their growth, and also for the plant growth promoting. So several kind of the microorganism, they have some just like the hormone or to contribute of the growth of plants. So TISTA also have done the research on the biocontrol, just like the production of the bioproduct to be pesticides and the plant pathogen. So we have the project right now in our demonstration plant to produce of the Bacillus thuringiensis, and Trichoderma is a fungi, and Beauveria and Metallysum to use at the bioproduct to agriculture to control of pest, pesticide, and plant pathogen. Next slide, please. The second application in food production. So the microorganism, they can produce the organic metabolite, just like the enzymes, organic acids, and they can play the major role to develop from the raw material, different kind of raw material to be the different kind of food product, just the typical flavor, typical texture, and also they can have more nutrition. For example, when we use the milk to be the raw material, we can use several of kind of microorganisms, just like the lactic acid bacteria to produce yogurt. We use the lactic acid bacteria, fermented milk, kefir, and we use the cheese. Also, we use the lactic acid bacteria, but the different species can be produced, just different kind of the food. Next slide, please. Also, the vinegar is really now in the commercialized, is the high value in the industry. So we use the fungi aspergillus to hydrolyze the starch into the sugar, and we use the yeast to fermentation of sugar to alcohol, and then we use the bacteria to transform of the alcohol into the acetic acid bacteria, into acetic acid. So it's the sample, and the bread, we very well known. We have bread almost in every morning. We use the baker yeast of Saccharomyces BCE. That's the main microorganism. They have to make the starch to be the bread. And the kimchi is traditional, the Korean fermented vegetable. So this also we use the lactobacillus of lactic acid bacteria to produce the lactic acid to preserve the vegetables, and can be the make the vegetable is soft and really good flavor. And also they have the ability just like to help ability something. Next slide, please. It seems there is a sample of the we use when we use the different kind of the raw material. When we use the soybean and the sauce, we can produce many kind of the various food when we use the different kind of microorganism. Just like the soy sauce, we use the fungi to hydrolyze of the soybean and can be digested into small molecule of protein, just like the amino acids. That is very helpful for the consumer, and we can preserve of the product as well. The natto is the traditional Japanese fermented soybean. So that use different kind of the bacteria. So make is the typical texture is sticky and typical smell. So make is by the Bacillus subtilis, and the tempeh is the Indonesian fermented soybean, is the high protein fermented soybean that traditional in the Indonesia. That's can produced by the fungi of the Rhizopus oligosporus, something like that. The next slide, please. Also, when we change to the raw material in the meat, we can make a soy sauce to ferment the meat to preserve. In Thai, we call sometimes we call nam or cycle, something like that. It's also used the lactic acid bacteria. And when we use another raw material, and we can produce just like the beverage of the beer, we use the beer yeast and Saccharomyces carbogenesis, or we produce the wine from the fruit juice, we can use the yeast of Saccharomyces cerevisiae. Next slide, please. The third application of the energy. So ethanol production. So we use the microorganism to produce the ethanol from the agricultural raw material, just like the cassava, sugarcane. So we use the enzymes, and we use the microorganism to convert the sugar into the ethanol, just like the yeast. Very famous, we use the Saccharomyces cerevisiae, and sometimes we use the bacteria of Pseudomonas mobilis. TISTA is the first institute in Thailand to do the research of the ethanol production for the vehicle. So TISTA has constructed the pilot plant of ethanol in the 1984, so it's almost 40 years ago. So, and we try to produce the ethanol and try to blend with the gasoline to be the new products of the gasohol, and we test run to the car and vehicle nearly 40 years ago, and we succeed that right now. Next slide, please. The energy, the biogas. So we know that when we have the water treatment system in the factory, so the wastewater can be utilized to be the energy to subsidize in the plant utilization energy that we use of the consortium of bacteria. So mainly is the methanogenic bacteria to produce the methane gas. They can use apply as the biogas. Next slide, please. The fourth application in the industry. So the famous metabolite of the microorganism used industry, that's the enzyme production. So the industry, the needs of the enzyme to process, to convert many raw material into the metabolite or to the finished product, just like the hydrolyte of starch by use the enzymes of amylase. Really huge industry to application of the enzyme amylase every year, and we convert to many kind of the variety of the products, just like some medical, some the energy, some of ethanol, something like that. And also just like the cellulase, protease, molecular and invertase also that can produce by various kinds of microorganisms, and then it can the application in the different application in the product. Next slide, please. Amino acids is famous that have reported that the world production of the amino acids around the world is about 500,000 tons per year, and nearly 95% in the top three of amino acids is the glutamic acids, methionine, and lysine. The three top amino acids is produced by using of the microorganism for the fermentation and use the commercialized process. So the glutamic acid is know very well, be produced by the bacteria and used as the monosodium glutamate that we can use for the flavor enhancer, and also the methionine and lysine used for the feed additive. Also use the different kind of bacteria to fermentation and produce the amino acid. Next slide, please. The fifth is the wastewater environmental application of the wastewater treatment. As mentioned earlier, that's the system of the wastewater treatment. We use the microorganism to degrade it or to digest of the biomaterial in the wastewater, and to reduce that bioorganic matter and make the water is more clean and can be released to the environment. Mainly we use of the bacteria as the main function. Next slide, please. Also in environmental application for the carbon dioxide capture from the release of the carbon dioxide in the surface of the world. They have to mention that the microorganism, the algae, is more effectively to absorb the carbon dioxide from the air. Is about 50% of the absorbed carbon dioxide in the world was absorbed by the algae, and answer 50% may be absorbed by the tree from the mainland. So it's very interesting that microorganism, microalgae can be held in the environment application. Next slide, please. Also, when we have the residue of the heavy metal in the environment, in the soil, in the water, something, just like we have the copper, chromium, or the nickel, something is contaminated in some soil. They make is really harmful to the environment, to the plant, to the ecosystem. We can use different kind of microorganisms, just like the bacteria, moss, and also of the algae. They have ability to degrade it of the heavy metal to be the natural and can clean the soil and the environment. Next slide, please. Also, when we have found the phenomenon of the oil spill or oil leak in the seawater, in some of near the seashore on the land. So they have reported that the microorganisms, they have ability to hydrolyze of the petroleum hydrocarbon as well. So mainly is a group of the bacteria. They have ability to hydrolyze the residues of the oils spilled from the water or seawater and can make the water can be clean and naturally is remediation. Next slide, please. So the group of the application of microorganism of the human health application. So very famous now, we use the microorganism is the beneficial microorganism we call the probiotics. It's the live microorganism when we ingest into our body and we will stay in our digestive gut can be produce the benefits to the host, to our body as well. For example, can inhibition of the pathogen and the reduce of the cholesterol or the improve of the immunity and also help about the digestion and also the absorption. So it be good for the host cell and can sometimes it can produce of the some vitamin and also they have ability to report that they can reduce of the cancer as well. So we can use this kind of microorganism to be generate of many kind of health product, and now it's very worldwide is commercialized in the industry in around the world. That's a very big and huge of the value in the market now. Next slide, please. Though antibiotic is just a bio substance biometabolite of the produced by the microorganism that can to be used just like the medical treatment to inhibit of the pathogen that infect of the human body. So many kinds of the antibiotics was reported and produced by the microorganism, different kind of microorganism. The first antibiotics was found in 1928, nearly 100 years ago, produced by the fungi of the just as Penicillium annotatum. The scientists of Alexander Fleming was found that and can produce that have reported as the first antibiotic found in the world and used for the human to make just like to treatment and curing of the pathogen. Next slide, please. Also, some vitamin really important to our body, and they have reported that some species of microorganisms they have really high potentially produce vitamin. For example, the vitamin B2 and vitamin B12. This is used the microorganism to produce for the commercialized now. And when we have some kind of the microorganism, for example, the vitamin B2, they use some fungi or mold to be the main microorganism to transform the raw material and produce the vitamin B2. And also vitamin B12, we can use the bacteria. So maybe in the species of the propionic bacteria, really famous and can use right now in the industry scale. Next slide, please. So the conclusion, we found that are some sample of the application of microorganism. So we found that the microorganism is mostly important, just like the topic that is really small, but it's really big, it's giant to contribute and to impact of our humans and to the world situation. So we try to live with the microorganism wisely and harmoniously and take care and save of their home and the world for our the happiness and sustainable for the human life living. Next slide, please. Thank you very much for your kind attention. Thank you very much.
Thank you, Dr. Bandit. Very interesting about microorganism study. I know that you have been done a lot of work on microorganism. I think the audience can have the question. Once again, if anyone has any question to Dr. Bandit, you can put in the Q&A box or the chat box. Thank you very much for the session today, and welcome back to join us again during Q&A. So I'm moving to the next presenter, which is from BCRC or Bioresource Collection and Research Center under FIRDI. We now have Dr. Sung Yuan Xie. He is the director of BCRC FIRDI with us today. So the topic of Dr. Xie will be a little bit on moment, please. The topic of Dr. Sung Yuan will be on research and application of microorganisms for food. Bioresource Center underpinning the bioeconomy development in Taiwan. Dr. Xie will share with us. Can I have a very short brief about Dr. Xie? He is also the president of the Mycological Society of Taiwan, and he received his PhD at School of Biological Science, University of Portsmouth from UK, and master and bachelor, another bachelor from the Department of Plant Pathology and Entomology, National Taiwan University. His areas of expertise are microbial biodiversity and application, mushroom biology, mycology. So please welcome Dr. Xie to cover on research and application of microorganisms for food. Please, Doctor.
Okay, thanks for your introduction, and good morning, everyone. It's my honor to give a talk in this joint webinar of virtual to talk about the role of BRC in bioeconomics. I will show our own experience in this presentation. Here is my outline of my presentation. And we know the microbiome exists all over the world, and they are small but important to our ecosystem, and also great influence to our life. The well-known story is the invention of first antibiotic, penicillin, was found from a small fungus, Penicillium notatum, in 1928. This invention saved millions of people's lives. However, some of fungi has been used for a long time. For example, the soy sauce made by Aspergillus oryza, it may happen 1,000 years ago. And except the fungi, actinomycetes is also an excellent source for searching the antibiotic, especially in recent decades. The drug resistance has become a serious problem. Searching for the new type of antibiotic were urgent. Bacteria are so small, but exist everywhere in the world. Some bacteria has been developed as a probiotic product. It takes over the major part of the healthy food market in Taiwan. It's about 10 billion NT dollars. Recently, the next generation probiotic like Academia mencinifera attract people's attention, may create another profit market. Algae is another superstar for healthy food. Not just produce oxygen, also could be the important source of protein, biofilm, and beta carotene. Yeast is so important for our daily diet. Without it, we can not taste the delicious bread, wine, and beer. Many viruses and bacteria, even fungi, could be pathogenic. However, they are also important bioresources for the researcher to make the cure. So where we could get those wanted bioresources, where we could keep them safely, a culture collection center or BRC could be the best answer. Here is the microorganism life cycle. The microorganism could be first found in the nature. After study, it is worth preservation. It may send to culture collection. After proper progressing, could be stored in liquid nitrogen for long-term preservation. When necessary, could be recovery and transport to the user. A culture collection is a depository of a microbial material, and it could be play a capacity industry role and to help country better understand their utility and their microbial diversity. However, the Biological Resource Center is could be a powerful organization. They consist of service provider and the depository include living cell, genome, and some informatics. BCRC is one of the department in FIRDI, which is a Food Industry Research and Development Institute. And FIRDI is a non-profit organization for the development of Taiwan food industry. BCRC is the largest division in FIRDI. BCRC is the only multifunctional BRC in Taiwan. Our mission is to collect and preserve and distribution various bioresources and provide the microbial professional service. We consist of 11 working units and 4 labs. Each handle the different kind of bioresources. However, different from the other culture collection. We have another 4 labs work on the microbial exploitation to create and edit their value. BCRC is professional and provide a service of microbiology. We create our own core technology in order to provide high value service for our partner. One of our core technology, preservation, we had developed the best preservation method for bacterial, fungi, mushroom, algae, animal cell, and the plant cell. For most of strain, we use the freezing dry and ultra deep freeze in liquid nitrogen or stored in -80 freezer. We also make a copy system in a different city far from the headquarters, about 180 kilometers away from our headquarters in case of some natural damage. Here is the general workflow for strain preservation. Normally, we receive a strain. We check the authenticity and the purity, followed by DNA sequencing to check the name of the organism is correct or not. And then we made a quality control and into the preservation bank. If necessary, we recovery to user. Till now, we hold more than 50,000 microbial strains and 14,000 cell lines and 1.2 million chromosome genes and bioresources for public deposit. We also provide the preservation service for private company. Till now, we service up to the 370,000 vial for for 390 company. A total of more than 34,000 strain. Most of strain is bacterial and filamentous fungi, and followed by yeast and mushroom strain. We distribution about 4,000 to 5,000 strain each year, and from the beginning of BCRC till now, we already delivered nearly 140,000 strain to the customer. For quality, our managed system certificate to the ISO standard from the year 2000. To provide high quality reference material, BCRC also meet the criteria of ISO and become a qualified reference material producer. The identification is one of the most important core technology in BRC. All material kept in BCRC have to check the name is correct or not. We have experts in taxonomy work on fungi and bacterial, actinomycetes, and algae. The identification technique has been progressed from the traditional to modern comprehensive technique. For example, from the morphology to API, the fast diagnosis kit, and followed by the DNA sequencing. And recently we built the multi-top technique. For fungi identification, the morphology is still very important, but DNA sequencing has become the essential and the standard method in BCRC. However, how to judge the correct name you blast from MCBI, sometimes it will be difficult to people lacking the training. Barcode system means using a specific gene sequencing to identify the microbiome. The barcode project keep running in BCRC. Till now, we complete our barcode about 12,000 strain and build a barcode system database for very faster to very useful to strain authentication. Last few years, we introduced multi-top mass spectrum system for rapid analysis and quality control for microbial batch production and record identification. We also built in-house MS database up to 1,290 strain, contain about 590 species. Will be very helpful for regular diagnosis for environment contamination and so on service. For important industry microbial species, like bifidobacterium, lactobacillus, and so on, we use the core genome multi-locus sequence typing method. It could distinguish microbial to strain level. We combination all identification technique. We complete more than 1,500 contract case for industry customer last 5 years. Besides the identification service, we have provided another more than 160 service to our customer. And among of them, 14 test items meet the ISO 1725 standard. We carry out about 1,500 case contract service every year for industry and academic customer. And here is a certificate for ISO standard. Through our website, you could find the strain information and various service information. We provide customer could order strain from the website online. Microbiome is a hotspot. It has been proved influence human health. We had isolated a large amount strain from the healthy adult and animal by using cultureomics platform. At least 90 new species has been acquired. From the human, 61 novel species has been found. And those, some of them, it could be developed as a next generation probiotic product. We also drive the traditional food to modern function food. For example, the red rice is a kind of traditional food. We start from the complete the whole genome sequence and decoding, followed by functional food study, metabolite study, and the novel function compound. Till now, we found at least 30 new compounds and has been published, and some of them are patent. We find some new compound could be anti-cancer and also anti-alopecia. The anka is made from the red rice. We help some company to make some new product and also have a lot of patent from the molluscus inverti. We are engaged in the collection of Saccharomyces yeast in Taiwan, and we acquired 230 strains from various environments and screening for some strain suitable for the bakery industry. We study the characteristic of yeast strain and produce a database. Strain with various features have become the superior source of yeast starter. Some special strains contain a higher proline and glycerol and trehalose with higher tolerance to freeze stress have been obtained and could be developed for freezing thawed baking yeast. BCRC also built a large cell resource bank containing various tumor cell line and stem cell. The request for cell line and service is increased up to 2,000 vials per year. We also provide a GTP level facility and get a good quality and safety test service for people running cell therapy. For a worldwide trend of alternative to animal experiment, we built two kinds of service and acquired GLP certificate last year could provide a high quality service. Since 2015, BCRC also started providing training course of photosynthetic bacteria, PSB. Till now, over 1,050 training. Most farmers, they apply the PSB to agricultural land, animal husbandry, and water product. This kind of promotion of the application of PSB is benefit of promotion plant growth and improve the quality of agricultural waste, water, and also help to the development of sustainable agriculture. BCRC is a continuously improve the technology in microbial identification, isolation, and cultivation, as well as maintain the function. As a BRC, we are eager to explore the possibility of bioresources application and provide the best service to our customer. BCRC make effort to harmonize the concept of the best practice of BRC and continue to upgrade for the requirement of the management technique level and quality of service for bioindustry. Here's my presentation. Thank you for your kind attention.
Thank you, Dr. Sear. So this is another interesting topic on the study that done at BCRC. So we will come back with the question and answer in the next session after the last speaker. So thank you, Dr. Sear. So right now we are going to have next session by our Thai researchers from TISTR or TISTR. So this will be from lady. So she welcome Dr. Nasisorn Suksi Deshasin. She is going to speak about probiotics, precious life inside us. So the topic is quite interesting to know more about what is inside our body from what Dr. Nasisorn has done the study. A little brief about Dr. Nasisorn. She is the expert in area of gastroenterology, intestinal absorption, epithelial transport, and cellular physiology. So right now, Dr. Nasisorn is with us here. So welcome, Dr. Nasisorn, to brief us about Probiotics 101. Precious life inside us. Please, Doctor.
Okay. Ladies and gentlemen, let me welcome you to my session. My name is Nasisorn Suksi Deshasin. I'm a researcher at Biodiversity Research Center. Today, I would like to talk about probiotics, precious life inside us. A recent study says that you have like 300 trillion bacteria living inside you. Much of these bacteria reside in your gut. The complex community of microorganisms in your gut called gut microbiome, which bacteria comprise the vast majority. Surprisingly, the metabolic activities of your gut flora perform important functions in the body, such as production of vitamin, help digestion, stimulate immune system to fight against infectious pathogens. So maintaining the high level of gut bacterial diversity is a key to maintain good health. A loss of bacterial diversity in the gut results in a physiological change within our gut system that can initiate common infection and inflammation. Unfortunately, the microbiome of the gut can decrease with age. That's why we have to focus on microbiome in the gut, just because it can be modified by yourself. Here is an example of a good bacteria. It would be the bifidobacterium, often found in fermented food, such as yogurt and cheese. And here is an example of the bad one, such as Salmonella, Clostridium perfringens, Campylobacter, and pathogenic strain of E. coli can cause foodborne disease or, as you know, food poisoning. Staphylococcal disease involved in skin infections. So disease occurs when the good bacteria are no longer able to control the bad bacteria. That means when your body doesn't have enough good bacteria, the bad one can thrive. Food and Agricultural Organization of the United Nations and World Health Organization have been further clarified the definition of probiotics as live microorganisms that, when administered in adequate amounts, confer a health benefit on the host. Probiotic foods including yogurt, kefir, kimchi, or fermented milk. Different types of probiotics may have different effects. For an example, if a specific kind of lactobacillus helps prevent some illness, that doesn't necessarily mean that another kind of lactobacillus would do the same thing. Therefore, choosing the right type of probiotic is essential. And those strains must have been characterized as generally recognized as safe or GRAS status. This is FAO's WHO guidelines explain various screening sections involved in the evaluation of probiotics for food, which our institute has been followed. This guideline, there are certain expectations required to call an organism as probiotic clearly must be included. The strain must be identified genetically and phenotypically. Screening of probiotic strain should consider the following aspects. The probiotic strain should be safe, free from contamination, non-toxicogenic, and unable to transfer the resistance to antibiotic to other kind in our intestine. In functional aspects, probiotics should have a great capacity to survive under intestinal conditions such as resistant to gastric juice, bile salts, and digestive enzyme, antimicrobial activity against potential pathogens, adhesion to our intestinal cell, and any other kinds of effects that have benefits on our health, such as anti-cancer, antioxidants, which specific to individual strain have been considered. And that microbes must be alive in an adequate number when administered and during the storage. Common dosage range are from 10 to 20 billion colony-forming units per day for adults. And growth efficacy of the probiotic upon technologic treatment should be demonstrated. Moreover, the ability to target the colon for the enhancement of gut microbiota to show their beneficial effects have been considered. All of these potential characteristics should be retained after encapsulation with cell protective agents. The next level is in vivo or animal study and human study should be delicate design. Then we develop them as a food and declare the detail of probiotic strain viable, bacteria count, shelf life, and storage condition. All of the aspects that I've mentioned can be performed by our skillful in-house team and provided as a service too. The most common probiotics are a member of lactic acid producing bacteria or LAB, which are able to produce lactic acid by sugar fermentation. Most of bacteria belong to a group called Lactobacillus and Bifidobacterium. Other kinds are Streptococcus, Enterococcus, and Bacillus. And so may yeast such as Saccharomyces boulardii right here. And here on the right is the example of our probiotics that has been manufactured by us. Here is an increasing evidence that probiotics may improve intestinal health and can help the microbiome rebalance after illness or treatment, such as antibiotic-associated diarrhea, decreased constipation. Moreover, the inflammatory bowel disease or IBD or a leaky gut wall can cause inflammation of along the digestive tract or gut lining. It can cause abdominal pain, diarrhea, fatigue, and weight loss. Several studies have found that those friendly bacteria in the gut help to strengthen the intestinal barrier and reduce those inflammation. Mention the immune system. Probiotics may give your immune system a boost and inhibit a growth of harmful gut bacteria. Natural killer cell, as you know, as NK cell, is the effector of our immune system that can directly kill the cell undergoing malignant transformation or tumor cell, and cell that infected with either virus or other intracellular pathogens. These guys are stimulated by antigen-presenting cell, mainly we call it dendritic cell right here. Dendritic cells are activated by interacting with commensal or pathogenic bacteria. In this case, we mentioned lactic acid bacteria, resulting in NK cell production of like interferon gamma and TNF alpha, which like cytokine that have been produced. This stimulation overall increase in NK cell activation and their cytotoxic activity. Showing right here in the blue box are our powerful strain for this effect, which produced by us again. The next topic is obesity that can be caused by dysbiosis of gut microbiota, as you see on the right side here. This dysbiosis can lead to the process of the decrease of the process of breaking down fatty acid in the liver and muscle, increase in fat storage, disrupt the host response to insulin, and cause more hunger, development of inflammation in adipose tissue as well. Surprisingly, probiotics increase the variation of intestinal microbiota right here. Inhibition of growth of pathogenic pathogen in our guts, stimulate short-chain fatty acid production in the gut, as you see right here. Probiotic can cause a healthy effect, as you see on the left here. Increase the process of breaking down a fatty acid, reduce of adipocyte size, and reduction of fat cell, help you feel fuller longer, burn more calories and store less fat, improve insulin sensitivity and increase glucose absorption. And as you see shown right here, a powerful strain of this effect that we have been discovered. Here is the article published by Tister staff, which said the effects that I have mentioned prior to this. A number of large studies have shown that certain probiotics may be able to lower cholesterol in our blood, particularly in people with high cholesterol level. Probiotics, they can bind with cholesterol in the intestine, as you see right here, and stop it from being absorbed in our body. The cholesterol is then excreted through our feces rather than stored in our body. Shown here is our powerful strain that we have been discovered again. In the past decade, research has shown that the gut and brain are connected in a system called gut-brain axis. Researcher indicates that probiotic in this loop, or we call it as psychobiotics, can help treat cognitive and neurological disorder such as autism, Alzheimer's disease, and Parkinson's disease. A good bacterial probiotic is capable to increase neuroprotective molecules such as BDNF right here, and resulting in improvement in cognitive and have a benefit into our brain. There are an example of our probiotics that we found to have this effect. It's Bifidobacterium longum and Lactobacillus rhamnosus right here. Recently, more studies show that probiotic may exert skin health promoting effects such as improved atopic eczema, atopic dermatitis, and healing of burn scars, skin rejuvenate properties, and improving the innate immunity. As seen in this figure, bioactive form from probiotics for dermal application, each compound that have been produced by probiotic have a protecting effect on each skin layers in various way. This effect was catch our attention too, and we have already planned the project about skin issue. COVID-19 is a huge topic right here. It's known to damage the body's immune defense through a cytokine storm of excessive inflammatory cytokine. This is believed to be the main cause of deteriorating health and even death. Because intestinal microbiota has been shown to strengthen the immune system and fight inflammation, researchers think probiotic supplements may help speed recovery from coronavirus by inhibiting of cytokine storm. It has been reported that several probiotics, particularly lactic acid bacteria, are known to produce bioactive peptides as an ACE inhibitor, where the corona pathogen enters our body to invade the intestinal cell. Also, people with COVID-19 have reported gastrointestinal symptoms such as diarrhea, nausea, vomiting, or abdominal pain, and loss of appetite, which probiotic can cope with. Research is in preliminary and much more data and clinical trials are needed, but it's very interesting. Here show the probiotic market size is expected to reach approximately 111 billion USD by 2030 in the future. The considerable production and consumption of probiotics are complemented by innovations in probiotic by key players, most notably in countries such as China, Japan, and India. From regional insights, our Asia Pacific led the global market with a revenue share of over 40% in 2021. This region is witnessing a substantial rise in consumer awareness due to competitive strategies adopted by the global players. Strong demand from countries such as India, China, and Australia is contributing to overall growth. The rising population coupled with rising disposable income and improving the standard of living is also expected to cater the regional market growth. On behalf of Innovative Center for Production of Industrial Use Microorganism, or ICPIM in brief, with the production rate of 25,000 per year or 1,000 kilogram dry weight under the system that prepare to be processed under GHP and HACCP system. We here to share the probiotic manufacturing process as teacher. Although the probiotics fall in the dietary supplement category, manufacturing them might be differ from the usual. Let's see how. The first step, starter preparation. Seed stock, which has been carefully prepared to consist of a single pure strain and verified to be free from contamination by quality control testing, is used in a limited number of sequential or gradually scale up seed fermentation to achieve the desired inoculum volume, and is ultimately transferred to the main fermentation vessel for growth. The next step is media preparation. The heat-treated medium used in the seed scale up and main fermentation is a blend of water, nitrogen source, carbohydrate, salts, anti-foam agent, and micronutrient necessary for the growth of probiotics. Moreover, every step, every portion, and every vessel must be sterile. Step 3, fermentation. Starter was inoculate to sterile fermentation media in the main tank. The fermentations are carefully environment and duration control. Quality control testing were performed during the fermentation step. Once we've achieved the desired growth, we will move on to the next step, which is separation. After the fermentation in the main tank is complete, the cells are concentrated by separating the cell from spent media through centrifugation. Depending upon the final product application, stabilizer solutions such as cryoprotective agent, which is the protected the cell from injury during freezing, or lipoprotectant to protect cell from injury during freeze drying, may be added to the cell prior to freezing step. The fifth step is drying. Once the probiotic concentrate is blended with those cryoprotectant solution I've mentioned before, various freezing process can be applied. One simple freezing technique consists of pouring cryoprotect concentration into a cans and immersing the sealed cans into a liquid nitrogen bath. Freeze drying length vary as a function of the strain or its formulation and the freeze drying cycle, but usually takes a few days to be complete. And the final step, packing. After removal from the dryer, the lyophilized material is milled to a powder, with defined particle size and density. The blend is then used to make finished formats such as a capsule, a sachet, or a tablet. Quality control testing is performed on in-process samples and the final product to make sure that the end product is high quality and free from contamination. And here is our real production unit showed. Here is our result from effective and powerful production unit in ICPIM that can produce high quality end product, as you see on the right side here. And the left side, there are an example of probiotic strains that has been picked up and changed into our product. Depends on tolerance capacity to technological process each strain have. So all the aforementioned health benefits should encourage investigators, companies, stakeholders, and consumers to learn about the effects of probiotics as a whole and evaluate those strains that show promising results. Even though there was a small, tiny organism, Biodiversity Research Center of Tista have been appreciate their powerful potential. Together, we have all potential from the reliable production process, the safety and quality control system, and efficiency characterization and analytical and testing until we can deliver our high quality probiotic products, which have enormous benefits and suit consumer requirements. So that's all of what we do here. Thank you for your attention. If you have any question, I will do my best to answer them.
Thank you very much, Dr. Nasisorn. So there will be the question and answer session after the next presentation. I will invite you back again. Once again, thank you for very useful presentation, and definitely Tister is looking forward for our audience here to visit Tister to see the ICPIM facility. That is very interesting and innovative facility in Thailand. So the next speaker will be the combination of two speakers on the technology transfer of microorganism research. There will be altogether 2 case studies, which will be the last session for this morning. So I would like to invite 2 of our speakers, which is Mr. Hing Yun Chen, Senior Research Scientist from BCRC under FIRDI, and Dr. Yang Sheng Kua, Research Scientist from the same organization. Dr. Chen graduate Master in Agricultural Chemistry Department from National Taiwan University, and he is expert in food science and technology, applied microbiology and probiotics. And another speaker, Dr. Kua, he received PhD from Institute of Molecular Medicine at National Tsinghua University. He is the expert in genetic engineering and microbiology. So there are 2 case studies to share here. The first one, lactic acid bacteria from microbial identification to market. And the second one is photosynthetic bacteria farming application. Right now, ladies and gentlemen, I will bring you to the video of the 2 scientists, and we will be back to meet both of them at the Q&A session. Please.
Ladies and gentlemen, I'm glad to have the opportunity to share the topic about lactic acid bacteria from microbial identification to market. My presentation will be divided into 4 parts. Starts with the marketing of lactic acid bacteria, and I will talk about BCRC30. We provide the one-stop service for the LAB industry. The patent properties, strengths of 30, and conclusion. The global probiotics market is expected to reach $105.63 billion by 2028. The consumption of probiotics has been rising due to the increasing disposable incomes of consumers and the growing trends towards consuming healthy and nutritional food. The spread of COVID-19 rose consumer awareness of nutrition and preventive health care. The consumption of immunity-boosting foods and dietary supplements increased significantly after the outbreak of the pandemic. The market size of LAB products in Taiwan is over 10 billion new Taiwan dollars, about $350 million in 2021. As we all know, the applications of LAB could be very diversified. They can be found in drops, heavy foods, yogurt drinks, or snacks. It is also used in cosmetic products and some animal health products. In Taiwan, 30 leads the research and development on LAB. 30 provides one-stop service for industry, including technology transfers and service platforms like local LAB collection bank, LAB systematic identifications, functional assay systems, production process technologies such as fermentations and downstream process. These technologies created a solid foundation for the development of LAB industry. This slide shows some marketing LAB products developed from Verdi's technology transfer. They also acquired the heavy fruit certification by Taiwan's TFDA. On the other hand, we also developed traditional fermented food products like yogurt and pickles. Here, I would like to talk about our service platform for the LAB industry. First, we have established diversified local LAB collections in BCLC. In the special collections, which is not published list or in our catalog, there are more than 1,000 strains. This is an important source for developing LAB products. This slide shows the guidelines for evaluation of probiotic strains for food use. Correct strains identification is a crucial step in this process. In BCLC, we have set up a polyphase taxonomy identification platform, which combines traditional methods and pioneer technologies such as morphology, phenotypes, genotypes, mass fingerprint. Is the accurate microbiome, the identification of the LAB, important to product development? The answer is definitely yes, because it is the key for evaluations the values of the strain and biosafety. For example, quality assurance, product certifications, marketing, patent protection, and biosafety assessments. All the above requires proper microbial identification. We use the multi-taught mass spectrometrics to rapidly identify and discriminate the bacterial colony after isolation. The speed and reliability of multi-taught make it possible to use the technology in quality control of commercial probiotic products. We also use a series of platforms to analyze the character and screen the functionality of the LAB strains. The potential of an industry strain could be completely evaluated. Nowadays, genetic screening of functional and safety properties is necessary for industry use. We have also set up microbiome-based technology to complement other experimental approaches to help us understand the complex microbial communities in the intestine. A lot of effort has been put into developing the culture-based analysis to identify microbiome at species level. Production process is a key to bring LAB products to the market. Scaling up the production process is usually a challenging task. BCLC 30 has examined specific needs and research issues that could be encountered in scaling up from laboratory equipment to pilot and full-scale industry unit. This slide shows the scaling up steps. from the Shake Fast 5-liter fermenter pilot plant scale from 20 liter to 2000 liter fermenter. Lactic acid bacteria are usually sensitive to temperature, which created stability issue and also affects the final trade price. A series of technology has been developed to enhance the stability of the product. In the spray drying process, we test different combinations of operational parameters and carries to protect the probiotics from inactivation to extend their shelf life. We established the microencapsulation coating technology to further protect the LAB products. Now, I would like to talk about the patent probiotic strains of Verdi. At first, one of our strains in BCG band was found to be able to improve the intestinal microflora. The total fecal and cecal bifidobacteria increased significantly in mice after LAB feeding. The same strains show the ability to enhance immunomodulation. In an animal experiment, the serum and intestinal level of the total IgA were significantly higher in LAB fat groups than the control group. Another LAB strain identified in Verdi shows the prevention of colony tumorigenesis by consumption of these strains. In male ICR mice, dietary consumption of these strains resulted in a significant inhibition of the DMH-induced ACF formations. Long-term dietary consumption resulted in a reduction of colon tumor multiplicity and the size of the tumors. The anti-cancer activity of the fermentation process was studied in an animal model. The growth of the solid tumor was inhibited after administration of the fermentation process as compared with the control group. In mouse model and other lactobacillus strains significantly decreased the total cholesterol level in plasma compared with that of mice fed with a high fat and high cholesterol diet. This results show that LAB strains could be a good candidate for reducing the risk of cardiovascular disease. With the advance in technologies, the development of the LB has gone through several stages. In the foreseeable future, the breakthroughs in the functions and performance of AB will lead to the fourth generation products. The food products that hit the market will be more diversified. In the medical sector, the LB products could be developed with certain disease as the target. As for genomic research field, comprehensive macro research analysis of genomics could be employed to study different aspects of LAB. With the increasing interest in probiotics and the rising awareness of healthy lifestyle, the advance in biotechnology will undoubtedly provide breakthrough solutions for future innovation in the ever-expanding applications of LAB. Well, this talk will end here. Thank you for your attention.
Thank you very much, Mr. Chan. So the next one, I would like to invite all of you to hear from Dr. Yang Sheng Guo, the research scientist of the BCRC FIRDI. He is going to share another case study. Please, Dr. Guo.
Good morning, everyone. It's my pleasure to report a farming application of photosynthetic bacteria. My name is Yang Zhenguo. Before introducing the photosynthetic bacteria, let's talk about the phototrophic bacteria first. By definition, photosynthetic bacteria are part of phototrophic bacteria. They can use light energy for their growth and metabolism. Some phototrophic bacteria, such as cyanobacteria, they are oxygen producers. They use chlorophyll to capture light energy and oxidize water molecule, then release oxygen. Another group of phototrophic bacteria called oxygenic phototrophic bacteria. They do not produce oxygen. They use bacterial chlorophyll instead of chlorophyll to capture light energy and use hydrogen sulfide as electron donor, so they do not produce oxygen. Most of photosynthetic bacteria is the anoxygenic. According to the morphology, physiological, and metabolic property, the anoxygenic phototrophic bacteria can be classified into 6 groups, including filamentous anoxygenic phototrophic bacteria, heliobacteria, acetobacteria, Gemmatimornidus, green bacteria, and purple bacteria. The FAPB can be further divided into red FAPB and green FAPB. The purple bacteria can be further divided into purple sulfur bacteria and purple non-sulfur bacteria. They are different from the tolerance to sulfur, such as hydrogen sulfide. The PSB can tolerate higher concentration of hydrogen sulfide. But today, I will focus on the purple non-sulfur bacteria because there are more and more literature discussing their ability to produce high-value substance and promote plant growth. PNSB is a phylogenetic diverse group of bacteria. They are classified in alpha and beta proteobacteria, including at least 6 order, 30 family, and 21 genera. The most studied genera are Rhodoviridae, Rhodobacter, and Rhodopseudomonas. The metabolism of PSB is diverse. In the illuminated and aerobic environment, they can use light energy to drive their metabolism. So they are phototrophic PNSB. And in the dark and aerobic condition, some PNSB can use chemical energy, so they are chemotrophic PNSB. When they use the CO2 as carbon source, they are autotrophic bacteria. And when they use the organic compound as carbon source, they are heterotrophic bacteria. So depending on the presence of nutrient, oxygen concentration, and light intensity, they can shift their mode of metabolism between photoheterotrophy, photoautotrophy, chemoheterotrophy, or chemoautotrophy. So the diversity of metabolism makes the PNSB widely used in farmland, aquaculture, and wastewater treatment. In recent years, we have collected a large number of phototrophic bacteria from soil, paddy field, wastewater, or marine environment. And after isolation and purification, now we have preserved more than 400 strains of phototrophic bacteria. And part of phototrophic bacteria are further sequenced and analyzed. Now most of them are PNSP in our library, including Rhodopseudomonas, Rhodoplan, Rhodobacter, and Rhodovilan. And most of Rhodopseudomonas and Rhodoplan are isolated from freshwater or soil. Then most of Rhodobacter and Rhodovilan are isolated from saline sample. For the application of PNSP, we designed a training course to teach the public to learn how to isolate and purify the PNSP. We charge for $500 for a 3-day training course. In a training course, they collect the soil or water sample from their farmland. Then we use a selective medium to enrich the photosynthetic bacteria. Then we teach them how to isolate, purify, and identify the PNSP. We also teach them how to prepare the culture medium and prepare the starter. And we also teach them a simple way to cultivate 20-liter PNSP. Here are the steps. Due to the anaerobic and phototropic property of PNSP, so we need a light transmitting and a sealable bucket. We add the culture medium and the PNSP starter into the bucket, then fill up with water and close the lid. Finally, we place the bucket in the sun or artificial light until it's turned red. As this photo shows, they put the bucket in the sun until the PNSP grows and turns red. If it turns red, they can use in their farmland or aquaculture. Here is a story. A retired minister of Council of Agriculture in Taiwan, Mr. Chao. He heard about this training course from a farmer, so he paid to learn this course too. After the class, he went around to observe the culture station of other students, or most of them are farmers. He think this training course is good for Taiwan's agriculture, so he encouraged us to apply for funding to run this course. And yes, we did it. Now the farmer learned this course is free. From 2017 to 2022, we have provided more than 50 training courses all over Taiwan, and we train more than 3,000 trainees. For the application in farming, the PNSP has the nitrogenase, which can convert the nitrogen to ammonia, so we can reduce the nitrogen fertilizer use in some crops. And some PNSP has the ability to dissolve phosphate in the soil, so which can increase the availability of phosphorus in the soil. And most of PNSP can produce PGPS, such as IAA and ALA, which can promote plant development and resist to stress. Some articles show that PNSP also have the ability of heavy metal bioremediation and greenhouse gas emission mitigation. This is a critical case of application. This pitaya farm is in Tainan. The pitaya is a long day plant, so its growing season is in summer. The farmer, in order to make it productive in winter, They were useful to extend the light to promote flowering. So in this trial, we use PSB combined with light to increase the number of flowers in winter. This line we used the PSB and this line was controlled. We can see the left picture. When we use the PSB, which can increase the number of flowers compared to the control group. And the right picture shows the root of pitaya. We can see the PSB can promote the root development. This is the nursery trial of cantaloupe. We soak the seedling with 100 flow dilute PNSB and the control group soaked with water. After 15 days, we can see the PNSB promote the development of leaf and stem. The length and the diameter of stem is significantly better than control group. For the application in aquaculture, because PNSB can reduce the hydrogen sulfide and ammonia concentration in water, so it can reduce the odor and the toxicity caused by hydrogen sulfide or ammonia. And PNSB also can produce many nutrients such as CoQ10, carotenoid pigment, and high amount of protein and vitamin, which can be used as bite or feed additive. This case is in Zhanghua. Mr. Li is a young man. He come back to help his father to manage the clam pond in 2020. And he also learned the cultivation method of PNSB and apply it in the clam pond. In 2020, before they learned how to cultivate the PNSB, so they did not use it in his clam pond. In 2020, they harvest 9,750 kilograms per hectare. The survival rate is 70. And in 2021, they used the PNSB to control the water quality, and they found it can reduce the ammonia and the hydrogen sulfide concentration in the water. In 2021, they harvest 12,000 kilograms per hectare, increase about 24% compared to 2020. And the survival rate also increased from 70 to 83%. In conclusion, we have collected a large number of domestic photosynthetic bacteria, and we will continue to enrich this kind of microbial resource. And we will continue to apply the photosynthetic bacteria through training course. In the future, we hope to develop another functional photosynthetic bacteria, such as mitigate methane emission, bacterial antagonism, or bioremediation. This is my report today. Thanks for listening.
Thank you, Dr. Guo, for very interesting on the technology that happening at your center. So right now it's come to the question and answer session. May I now invite all the speaker, Dr. Bandit, Dr. Seer, Dr. Nasison, Dr. Chen, and Dr. Guo to open the video camera to join the question session here. So I would like to put in a way that I am very much interested to have a question that come offline to me here that go through every scientist, researchers with me here. That the very interesting thing is definitely more food, less land. it is for sure that's happening with the biotechnology, bio-based. And there is a discussion on fermentation, and it is kind of traditional and trendy process to reflect our commitment to ecology, I would say. It's reflect to the BCG model and resilient economy. I would like to put this question to every one of the researchers and scientists here, that what is the challenge that you find in your work in order to develop microorganism, create the data bank, and what are really the challenge after your research to be done on the industrialize it in the food industry? So I go for lady first, Dr. Nasison. What you see the challenge that may happen with the technology that you are using to apply into the industrial food industry?
Okay, well, for me, the current role of probiotic researchers. So despite widespread studies, researchers are still challenged with identifying all the microbiota in the vast and changing microbiome in human gut. I mean, it changes all the time period. In the future, when we compare with in the past, it's not the same. And they also need to establish a system for standardize the research results to develop a clear therapeutic recommendations for probiotic users. That is for me.
All right, so I think the human organism also complex, and also the microorganism also complex. So the challenge is like, what is the consistent level, how we apply in therapeutic level to use those in the food processing industry. So come to Dr. Sear, Dr. Sun Yun Sear. So what do you think, sir, on the challenge that you guys facing?
Okay. The question is for service for foreigner. Is it?
Not really service for foreigners, sir. You have done a lot of study on the biotech fermentation and microorganisms that to extract it for using in the food processing. My question is, what is challenges you may, like Dr. Nasison answer her challenge is, sometimes the analysis and research say something, but human reaction when using it, it's change. That is the challenge she feel, she had. What about you, sir?
And we provide various service for different kind of customer. And so there are many kind of issue we have to resolve for our customer. And every year we provide about 1,500 contract case. And some is difficult. I think is for the government regulation is quite difficult to change. So the normal small company, they could afford to pay a lot of money to do the research. So we, through our effort, we could help them to overcome their technical problem and to produce some new product. It's our experience, yes.
Yeah, so I can say that some regulatory not even know the method to identify the pathway. So your organization also help the regulatory to determine the pathway.
Yes. Yes.
Okay. You have 1,500 cases. So mostly in Taiwan, or also serve the foreigners as well? For sure. Yes. Okay.
Some of them is come from the foreigner, especially they buy the culture from our stock, and we could deliver culture to the foreigner. Yes, no problem.
Okay, I saw that you have about 459 strains. So I think, ladies and gentlemen, you can contact BCRC if you are interested in any culture. They can be at your service. Okay, so I will ask Dr. Bandit on the. I know Dr. Bandit for quite some years, and he is working on the bio and fermentation plan. So Dr. Bandit, what is the challenge that you see the biotech products can be used in the food industrial level?
Okay. I say really have the good opportunity, very interesting now. So they have the research of the microbiology to apply in the food industry, especially in nowadays about the health products. So many industry they concerned about the health or something. So many kind of microorganisms that can produce the benefits to the human life, just like now we enter to the aging society in the world, in the Asia, in Thailand, and so on in the Asia. And also we be facing with the NCD disease, non-communicable disease. So many kind of disease in effect of our human lives, in the cancer, in the immunity, or some the Alzheimer's, something like that, diabetes, something. So we can apply the microorganism, just like previously that all the speaker has mentioned. It's very beneficially to use microorganism, and then the future is it can extend the research in another area that's close to the health product, and also in some futures that we predicted what is coming. For example, like the personalized, we can predict of the personal disease or personal the character of the health of each people, and we can design to use. So it's really a good opportunity that still remain of the big chance to use the biotechnology or the microbiology to apply in research and to apply in the industry.
I think you have addressed a lot on the benefit, a lot of concern on health and wellness, and even our country is aging society. You has addressed the area that it can work. However, I really want to hear from you, as you work in the research for long. What is really the challenge in terms of the implementation and application in the commercialized level from research perspective?
So the challenge now with the researcher, R&D, and the research institute or the university. So we have done in the level of the laboratory, and then we can to make it like the scale up or the demonstration plan, just like the TISTA. TISTA constructs like the Dr. Nasison has mentioned that we construct of the ICP for the center to production of the potential of some microorganism. So it's made to more the possible that not only the research in the laboratory only that can be applied in the commercialized scale. So this is the connection between the laboratory level and to the commercialized field. That's the research institute or the university have to close the gap by we can research in the make to the scale up and commercialized level.
All right, so I will come to the next round after everyone answer my first question. Dr. Nasison, you can prepare, because there is the question about if SME interested to use the service, how to do. Okay, come to the next speaker on the challenge facing in the using the biotechnology and development and microorganism and apply into the food processing. Still continue with the same question on the challenge. I want to hear from Dr. Guo. Dr. Guo, what is your learning and challenge that you have faced working with your consulting group or your clients to your center?
Hello. For our research, we identify a lot of phototrophic bacteria, and now we provide the training course for the farmer. Okay. If we want to continue to provide these kinds of service, we need to apply more government funding, and in the future, maybe we need to transfer this kind of technology to some company. That's all.
I think it's similar to every country. The financial support, government funding is kind of very important to drive the project. And yes, technology transfer for the private corporation to really make it into the bigger scale from the research you have done. Totally agree, sir. But the challenge is how we can communicate to the world that this is the work that has been done. This is a great opportunity having this webinar to share what's going on. So right now we come to the last speaker to share about the challenge. I saw from Mr. Chan that sharing a lot on lactic acid bacteria. This is very much interesting as well. How you identify microbia into the market, but I think you are the one that can answer more close to the market. Mr. Chan, I asked you about the challenge you have faced. Mr. Chan,
yes, I have developed many products to the markets, help to the small business or small company or the farmer. Also, we helped some company to make the probiotics products to the market, like the identification and scale up. So we have several products was certificated by to the health food by the Taiwan TFDA. This is very difficult to certificate in Taiwan. So we have so we have many products in the market now. Thank you.
All right. So I think Mr. Chan will be another person to share what is the learning like, because the consumer demand is very complex nowadays. We have to know how to position these complex products in the complex market. So right now I come to the next round of question will be like from the audience here asking, if I am SME, I want to really work for my products with the microbial-based products, health products. This question I would say to Dr. Nancy Son, if in Thailand, how is this growing opportunity, and is it difficult to work on this? Dr. Nancy Son, please.
Okay, well, I will go like Dr. Chan. We've struggled facing with Thai FDA too. According to the question, we have to divide them into two topics. First, if you mention to produce a microbial-based food but not health claim, or you need to prove it like safety, like are there any other pathogenic micro contamination in your product or heavy metal contamination along the production process. But if you need to put the sentence like this probiotic can decrease like blood cholesterol and on your product when you going to sell on the market. You need to prove any other aspect like efficacy of your product, viability of your product, or the living cell should be like 10 raised to the power of 6 or like the power of 9 along the shelf life that you have been claimed, like 1 year or 1.5 years. And about the growth opportunity in Thai market, it has been shown that 7% in dietary supplement growth even in COVID situation. So I think it's a best opportunity if you like to jump in this market. Thank you.
All right. So the answer is quite clear that if you are interested, it is opportunity for you to jump in, but to work in what application, what type of health food, you really have to work hand in hand with your researcher and understand what you want to position your products. I think every country face now is about health claim. So you have to really understand health claim under your country's FDA. This is I can grab from Dr. Nasison answer. So right now come to our other doctors. The question is, the microbial is really the help to reach the resilient economy or the bio-based economy. Is it the situation right now if we implement the microbial doing the biotech, studying on the biotechnology opportunity? This is at the timing that it can help the private company to recover from pandemic impact, or it's just about the reaching the resilient economy, being in the sustainable world. So I go to Mr. Chan first. Mr. Chan.
Yes, I'm agreed that the microbiome basis study is very complex. So we study the DNA-based approach, and then we use our technology to set up a culture-based approach to find the functional strengths. It's very important to develop the microbial food. Yeah. So on the other hand, the culture is very unknowable. It's very difficult to culture because it's very sensitive to the oxygen. So how to culture the strength in the production process is very difficult. Yeah, I'm agree. Okay.
Okay, understand. So I will have another question to Dr. Xie, Dr. Sung Yuan Xie. Sung Yuan Xie. Dr. Xie, as you mentioned about a foreigner, you also provide a service if foreigner interested in working on specific culture, specific strain from your microorganism bank at your center. What is the process to do? Is there any protocol to be done, sir?
And normally, the customer could access our website. And from our website, we provide the instruction about our service and the catalog of our strain. Then you could order from the website, and or you could send an email to our window of service unit, and we could contact. And normally it's for the strain exchange or for the strain you purchase from a foreigner, they have to pass our FDA some custom quarantine to check the strain is no harm, then we could pass the customer. Yes.
So the center also will help the foreigners to identify the process of how to take it out from Taiwan and grow in another country as well.
Yes, we could help the customer to dealing with the issue from the customer or from some quarantine bureau. Yes.
Okay, Dr. Xie, since you are here, can you share with us the highlight of the strain and products that's really the top one from your center in last 2 years?
The famous or the top selling is we call it Anthracia cinnamomea. It's a kind of medicinal mushroom, and also some bacterial probiotic strain lactobacillus is also very best selling, and some strains for test for the laboratory contamination is also very best selling from our culture collection. Yes.
All right, thank you, sir, Dr. Xie. So now I have another question, the last question to Dr. Guo, due to in ASEAN, we are, except Singapore and Brunei, we are pretty much the farmers. So I see that the case study on the farm application. My interest to know, like, Dr. Guo, for the farm application, can you share how you work with the farmers? Do your center spend time like Tister doing the area base, going to help farmers, educate them how it works from your center, please?
Thank you. We provide the training course, and we went around all over Taiwan. Then in the training course, we teach them how to cultivate the PNSB, and we teach them a simple way to prepare the culture medium. The culture medium is the select medium, and we teach them how to cultivate in 20-liter bucket. Then, because we are good for in the how to cultivate the bacteria, but we are not good in the farming. So we need them to we teach them to cultivate the PNSB. Then they use it in their farm. Then they can teach each other and how to use it in their farmland.
All right, I think this is another case study that audience here may approach to you, because most of our country are more on aquaculture, agriculture. This is really can reduce the antibiotic use in the ponds and can reduce the pesticide in the farm. That is kind of very interesting, but I agree, sir, we are researchers. You are good in bacteria, but not in the farming. So how to synchronize and like support and complement each other on the core area. Ladies and gentlemen, and all speakers, it's time for our break now. But before end up the morning session, I would like to once again thank you to all speakers here from Thailand and Taiwan. And before we go for lunch break, I would like to have a bit of introduction. Our top sponsor here, Vitro, which has created the hashtag on social media that we encourage and want to have the campaigns to promote everyone to have hashtag Waitrose event, Waitrose member outreach, Waitrose funding series, Waitrose new partner. And right now, I also would like to share on the video by Waitrose. After the video here, I come back again and brief about the afternoon session, please. All right, that is the video from World Association of Industrial and Technological Research Organization, Waitrose, the key supporter for this event. And ladies and gentlemen, the morning session, you know our expert speakers that is with me now. And we are going to have a lunch break and we will come back at Thai time 1:15 for the afternoon session. The afternoon session you will see with the award winners from the Waitrose Innovation Award winners 2021 as well. And I would like to promote all of you, if you are here, the good session like this can come again with the support of our organization together with me here and Waitrose. Put hashtag Waitrose event, Waitrose new member and Waitrose new partner. Once again, I will see you again at 1:15 Thailand time, GMT +7. And please don't log out from the Zoom. You can go and take a break and come back with the session 2. Thank you very much and enjoy your lunch.
Located in Nakhon Ratchasima Province, northeastern Thailand, there are the hotspot ecosystems covering an area of 78.08 square kilometers. The Sakerat Environmental Research Station, SERS. It was established by a resolution approved by the Cabinet of Thailand on 19 September 1967. Sakerat Environmental Research Station, SERS, is under the administration of the Thailand Institute of Scientific and Technological Research, TISTR, and the Ministry of Higher Education, Science, Research and Innovation, MHESI, of Thailand. The station has been designated by UNESCO under the Man and Biosphere Program, MAB, since 1976. Presently, SERS is recognized as the International Network of Biosphere Reserves, and being an administrative authorities of Sakerat Biosphere Reserve, the first biosphere reserve of Thailand, and one of 168 biosphere reserves in Asia and the Pacific region. Sakerat Environmental Research Station, SERS, and Sakerat Biosphere Reserve is located among the Dong Payayen Khao Yai Forest Complex, a UNESCO World Heritage Site, Taplan National Park and Khao Yai National Park, Khao Peng Ma and Pa Khao Pu Luang non-hunting areas. Sakerat Environmental Research Station, SERS, is located at the core area of the Sakerat Biosphere Reserve. Mainly contains two natural forest types, dry evergreen and dry dipterocarp forests, covering approximately 70% of the area. SERS is the flourished area to maintain high biodiversity, consisting of 196 plant species and 500 wildlife species. A significant species is mainland sirrow, a Thai-reserved species. According to the IUCN Red List of Threatened Species, over 17 threatened species natively inhabit the SERS. For example, Malayan pangolin, CR, elongated tortoise, CR, dole, EN, Asiatic black bear, VU, etc. There are 7 species endemic to Thailand, namely Miriam's legless skink, angulated bow-fingered gecko, and Korat big-mouthed frog. For those reasons, Sakerat Environmental Research Station, SERS, turns out to be a research center, a learning site for conservation and ecotourism, and an outstanding place for its abundance of resources and biodiversity. SERS has applied the knowledge from the research to achieve the readiness of station management and protection of the abundance of resources and the environment at the national and international levels. The other important missions of SERS are to transfer technology and provide knowledge to students, researchers, local people, and ecotourists, both in the country and overseas. SERS provides science camps all year round for the students and young generation, career training to local communities for sustainable living with nature, including services for ecotourists to experience the beauty of nature, which is building awareness of conservation on nature and environment. Here at Sakerat Environmental Research Station, we commit to retain a forest of learning for the wisdom of everyone, all sectors, and all nationalities.
Sawasdee ka. Good afternoon and good morning for our speakers and colleagues from Europe. My name is Rumpet Chitanuwat. I'm going to be the moderator for this afternoon session today. This morning, we have a lot of knowledge and expert sharing on the microorganism and utilization for food industry. This is part of the webinar today, small things but giant steps to biocircular green, BCG and resilient economy by Tister under food and wellness for the future and business opportunity. So Tister, the Thailand Institute of Science, Research and Technological is the host of the event with the support of World Association of Industrial and Technological Research Organization, or WITRO, and we also get the support from National Research Council of Thailand, WITRO, Vitro Bioresource Collection and Research Center, BCRC from Taiwan, Food Industry Research and Development Institute, FIRDI from Taiwan, Department of Bioscience, Horst University from Denmark, and Green Ant Company from the Netherlands, and Lechat Technological Center from Spain. You are with me on the second part of the webinar. Today, small things but giant step to bio-circular green economy. For today's program, we're gonna have another 3 speakers with me, which is in the afternoon session will be about the insects as a trend of alternative protein source. So let me brief you a bit of the agenda today. 50 minutes with the first speaker, Dr. Joachim Offenberg, on the global trend of edible insect as an alternative protein and future food source, linkage to SDG and resilient economy. And the second speaker on business opportunity with ant farming from the Netherlands by Mr. Mario Eduardo Simarco, founder and CEO of Green Ant Company. And the next one is the winner of Vitro Innovation Award 2021. We'll talk about the insect-based food source to supplement nutrient deficiency in vulnerable areas by our winner of the Vitro Innovation Award, Ms. Sandra Braussels, the R&D Senior Researcher. All right, so right now, I think everyone is ready to know more about the insect as the alternative protein and become the trend around Thailand, Asia, and even in Europe. Our speakers today are all from European countries. First speaker of this afternoon session, Dr. Joachim Offenberg. He is the senior researcher at Aarhus University. He focused on decade-long research on weaver ants. Being among the most known scholars in the field, he is based in Aarhus, Denmark, taking position both senior researcher and co-founder of Green Ant Company in the Netherlands. So I would like to welcome Dr. Joachim, who will speak on the global trend of edible insect as an alternative protein and future food source linkage to SDG and resilient economy. Please, Dr. Joachim.
Thank you very much for this very fine introduction. And first of all, thank you very much for inviting me to this exciting webinar. And also thanks to the organizers for arranging something like this. I mean, in Denmark, we are more or less over the COVID, all the COVID restrictions by now, but I think it's a very good idea still to do webinars like this. Because we can, in a cost-effective way, put people together from all over the world to share our ideas and inspire each other. So thank you very much for the organizers for doing initiatives like this. I would like to try to share my screen.
Yes, please, you can share.
Yeah, I will share. Do you see my screen, my presentation?
Yes, yes,
Yeah.
I see the ants already. Yep.
Okay. Good. So as you may have noticed by now, there's a slight change to the title. The title of my talk will be Weaver Ant Farming, a green technology for resilient farmer economies. And I'm using this title because I'm actually an expert in weaver ants. I've been working many years with weaver ants and how to use weaver ants in plantations. And I've also been working many years in Thailand on this. And I would like to maybe introduce weaver ants. Weaver ants are ants that live in the canopy of trees. They weave together the leaves so they can actually stay in the canopies, and then they can be highly abundant. You can have so many trees or so many ants on each tree. And there are a number of benefits if you learn about these benefits of the use of weaver ants. I call my speak green technology because the use of weaver ants is a sustainable measure compared to the alternative, which is the use of pesticides. And I also call it resilient for farmer economy because there are multiple benefits that can be achieved via the use of weaver ants. And not only benefits, but also multiple incomes from these ants. And therefore, I think it's resilient for farmer economies. There will be at least 3 different benefits that can be achieved. One of them is pest control. Maybe many of you know that if you climb a tree with weaver ants, then you will be attacked by these ants. And then you may also realize that if ants can chase away humans, they can also do the same thing with pest insects. And in this way, ants can be used for pest control in plantation trees. What may be less well known is the fact that ants can also, to some extent, fertilize their host trees. And I would also get into that during my talk. And then lastly, probably as many of you know, especially people from Thailand and Indonesia, will know that these ants can also produce animal protein, edible protein actually, because they breed ant larvae that are edible, both for humans, but also for livestock. And these ants larvae can be harvested and provide an income for farmers. But if we start out having a look at the first benefit, then we look at pest control. And pest control can actually be divided into 2 different types of control because ants, as I already mentioned, can control pest insects because they feed on other insects, including pest species. What might be more surprising is that ants can actually also, in some cases, control plant diseases. So I would also touch a bit upon this topic in my talk here. But if you start out looking at insect pests, then many farmers realize that their crops are attacked by pests. But if these trees are associated with weaver ants, you will see much less pest attacks on those trees. And weaver ants are especially effective in controlling pests because they are highly efficient in cooperating and communicating. When an ant meets a pest insect, it will call for help among the other ants. And in this way, weaver ants can knock down pest insects that are much bigger than themselves. So one of the reasons that ants are effective in this way is because they use communication via pheromones. And I would like to show you a small video. Here you will see a moth sitting on the ground, flapping its wings, but getting nowhere. And the reason is because there's a weaver ant that has bitten this moth. The ant cannot kill the moth, but it can hold it. And right now, this ant will be emitting an alarm pheromone. So all the ants in the neighborhood will now be able to smell that there's one of our nest mates that need help. And then sooner or later, they will find the source of this pheromone and they will show up at this site with the moth and help out this one ant. So sooner or later, here we see the next ant coming. Now this ant will also bite and also start to emit the same pheromone, making the signal even more pronounced. And in this way, ants can communicate and cooperate, and that makes them highly efficient in controlling insect pests. Here's another video from Tister's research station, where I were a couple of months ago. And again, you see that ants cooperate in knocking down, in this case, an hemipteran pest insect. So this kind of cooperation and communication makes these ants highly efficient. This is a photo from a mango plantation in Australia, in Darwin, Australia. And as you can see, these trees are connected with blue ropes. And this is because this plantation is protected by weaver ants. And there has been some studies done in exactly that plantation. For example, it has been estimated how many ants are there in the trees of these mango. And in each mango tree, it was estimated that there will be around 30 to 50,000 weaver ant workers. And under normal conditions, these workers are spread out throughout the canopy because they forage individually to cover as large an area as possible. So this canopy will be full of ants, like 30 to 50,000 ants. And that makes it very difficult for a pest insect to colonize this canopy without very soon being detected by an ant. And when it's found by an ant, this ant will call for help, and then all the ants would aggregate in that area and knock down the pest outbreak. And then the trees are connected with lines, because then ants can move to those trees where a pest outbreak is taking place. So weaver ants in a plantation more or less vacuum clean the canopies and attack more or less everything that moves, including different pest insects. And it has turned out that this method is rather effective. Two researchers, again in Darwin, in Australia, was trying to assess how efficiently are weaver ants compared to the use of chemical pesticides. So a Chinese researcher, Ren Kangpeng, and an Australian guy, Christian, they did 3-year studies in 2 different plantations, crops in Darwin. They worked at the same time in cashew and in mango plantations in that area. And then they tried to compare how efficient are ants compared to pesticides. And what they came up with, that in both cases, ants were able to control all the major pest insects in these 2 crops. And this is again one of the benefits with ants, because they feed on many different species, and they will even attack anything that moves around in their territory. As I told you, if you climb the tree, they will also attack you. So they are effective against many different pest species. In their studies, they also found that the ants were much cheaper to use compared to spraying chemicals, because the ants more or less take care of themselves. You have some costs during the first year if you want to transplant ants from natural environments and into your plantation. This would of course take some time. But after that, the ants more or less take care of themselves. You can manage them by feeding them to improve their production if you want to harvest ant larvae. But if you only want them for pest control, they are very cheap to use. And of course, the alternative will be buying expensive chemicals and spend time on spraying them several times during every season. So the ants are cheaper to use. And surprisingly, the ants were also more efficient than using pesticides, because in both cases, ants led to higher quality of nuts and fruits. So you actually have something that is cheaper and at the same time more effective. And even in cashew, you also had higher yields, of course, increasing your income. In mango, they had similar yields in the 2 treatments. But when the researchers put those numbers together in a cost-benefit analysis, then they came up with the result that in both cases, ants were able to increase the net income with more than 70% if farmers switched from using pesticides and using weaver ants instead. So there is a huge potential here for saving money. I mean, if you substitute the use of pesticides with ants, then you're actually substituting an expense with an income, because ants can produce ant larvae that can be sold. So the expense are the pesticides, and they will be substituted by ants that can provide an income instead. So there is a huge potential here. And I would like to point out that ants does not only function in cashew and in mango. They have many different host species. And I would just like to show you a table from a review here. We don't need to go into all the details of this table, but just to show you, for example, here's an example of citrus. Citrus was protected in India. A plus here means that it led to pest reduction. It also led to a damage reduction and to an increase in yields. But my point here is that from this review, we saw that at least 9 different crop species are protected efficiently in 8 different countries, and sometimes even Some of these studies even made a cost-benefit analysis showing that the ants were superior to alternative control measures in these systems. And you may also notice that we don't only talk about fruit and nut trees, but also timber trees like red cedar in this case here. So weaver ants are useful in many different types of crops. And actually we are running a program in Thailand at the moment where we would like to try out if they also work in, for example, avocado, jackfruit and durian, etc. But there is a potential to use them in many different types of crops. Yeah, so that was a short note on the use of weaver ants against pest insects. What I would also like to touch upon is that ants can be used against plant diseases. And it may seem strange, how can small ants protect whole trees against diseases? But one of the answers probably lie within the fact that ants live in very crowded societies. I have a small video here showing an ant trail, not a weaver ant trail, but in this case, a wood ant trail. But it would be the same with weaver ants. And you should notice that these ants meet each other within seconds. So you could imagine if one of these ants were infected with a disease, that disease would spread like an explosion in that society. So ants are challenged with disease transmission. And therefore, during ant evolution, they have evolved a number of measures to try to reduce disease transmission. And one of these measures is the use of antibiotics. On these photos, on the first photo here, you see that ants have a gland just above the third leg. This is called a metapleural gland. And this is a gland unique to ants. No other animals have this kind of gland. And the gland produces antibiotics, which are broad spectrum, effective against many different pathogens. And the ants use these antibiotics from this gland to fight ant diseases, entomopathogens. So the ants can produce antibiotics themselves. In other cases, we see that ants are covered in bacteria. This is a leafcutter ant covered in a white bacteria. So in this case, this ant grows a bacteria on its body, and then this bacteria can produce antibiotics that the ants can use against ant diseases, or in this case, against fungal diseases in their fungal garden. But the point here is that ants can produce antibiotics themselves, and they can also use bacteria to produce antibiotics to protect them against ant diseases. However, we have noticed during our field studies that we sometimes see reduced plant disease symptoms on trees with ants. So we were wondering whether these antibiotics had an effect also on plant pathogens, and if they somehow are deposited by the ants when they walk on the plants. So we first of all, we searched the literature to try to find out what have been done in this field. And in this first review that we published in Oikos, we found that we were looking for studies where the effect of ants have been tested on live plants against a plant pathogen. And we were able to find 30 cases, 30 combinations of ant species, plant species, and pathogen species. And out of these 30 combinations, we found that ants reduced the plant pathogen in 18. There were 6 cases where ants actually increased pathogen incidence because ants can act as a vector spreading spores around on the plants. But in the majority of these studies, we found that there was a pathogen reduction by the presence of ants. And also we did a meta-analysis to try to quantify how is the size effect of ants on plant pathogens. And we found that the effect on pathogens is not significantly different than the effect that ants have on herbivores, which was a bit surprising because, I mean, the effect of ants on pathogens has been more or less overlooked. No research groups have really been working on this systematically. and definitely not in an applied aspect. So to realize that ants might be just as useful against plant pathogens as they are against insect pests was a bit surprising. And we are very keen on this research project at the moment. So encouraged by these results, we did a second review where we were looking for researchers that has been trying to test different ant chemicals against plant diseases in the lab. So now we are talking about in vitro studies conducted in petri dishes inside the lab. And in this case, we found that we were able to identify from the literature 22 different ant chemicals that are able to inhibit at least 20 different plant pathogens that they were tested against. And on top of this, we also found that ants are associated to 30 different bacterial symbionts that again can reduce, in this case, 10 different plant pathogens. So there's definitely something going on between ants and plant pathogens. And these two reviews, of course, cover all the different ant species that we see around the world. But there was also evidence from the use of weaver ants in these results. For example, we have seen a study showing that a gaster secretion, a body secretion for weaver ants, are able to reduce soft rot caused by Pseudomonas aeruginosa. And this was an in vitro study, so this was an inhibition going on in a petri dish. We have seen from one of our own students at Aarhus University that crushed weaver ants can also inhibit apple brown rot caused by Monilia fructigena, again in petri dishes. And then we realized that hexanal is a chemical compound that weaver ants excrete as an alarm pheromone. And we found out that exactly this chemical compound is being used by the fruit industry to prepare fruits, and in this case, and in this way, limit fruit decay and increasing shelf life, because hexanal is inhibiting a number of different pathogens that leads to fruit decay. So weaver ants are excreting hexanal, which is used by the fruit industry to protect fruits. We also found a study showing that formic acid on mangoes will reduce mango skin rot and in this way increase shelf life. And this was an in vivo study comparing fruits from trees with and without weaver ants. We have seen that ants on orange trees can reduce anthracnose and green mold, again leading to increased shelf life, and again comparing fruits from trees with and without weaver ants in an in vivo study. And then lastly, there's been a study showing that ants on mango trees shows less damage by mango scab, and thereby also lead to higher fruit quality, as we saw in one of the previous tables, again comparing fruits from trees with and without weaver ants. So there's definitely a potential that weaver ants can also protect some of the plant diseases that farmers are struggling with in their plantations. And just before I leave this topic, I would like to show you some encouraging results, in this case, not from weaver ants, but from wood ants in Denmark. We are currently doing a project where we are testing wood ants as well as weaver ants. But we started out with the wood ants. And this is just to show you how it can look like, how effective this can be. These are 4 petri dishes with 2 compartments in each. The left compartment in each is a control, where there has been no weaver ants, and the left side are a compartment where 3 wood ants have been running around for 24 hours. And after that, they were inoculated with apple brown rot, a plant disease. And as you see in the left compartment, the control compartment without ants, there's a vigorous growth of sites. There's no growth at all. So the presence of ants in 24 hours has led to an elimination of this plant pathogen. There was another study done where a wood ant was running only for 10 seconds on a plate of agar. And as you can see here, they deposit like a trail of bacteria that are deposited and then afterwards grow on this agar. And one of the most abundant bacteria from these trails were isolated, and we found that it is a new Pseudomonas species, an undescribed species. And this species is extremely efficient against this plant pathogen. These are two control plates where there has been no bacteria. So you see white growth of the plant pathogen. But in all the remaining dishes, we have like 4 colonies of Pseudomonas, and they totally annihilate the plant pathogen. I mean, usually you will see a reduced growth, but in this case, you see no growth at all. So we found a bacteria on wood ants that are deposited where it walks within 10 seconds, and which has a highly inhibiting effect on a plant pathogen. So this is something that we are working heavily on. And if anybody among the researchers or other participants in this webinar is interested, and we will be highly interested in collaborating on this. We could work on wood ants in Denmark and weaver ants in Asia. Okay, so that was pest control with the use against insects and against plant pathogens. My next topic are taking a look at plant nutrients. We have realized that weaver ants can actually, at least to some extent, fertilize their host plants. And we noticed this when we, many years ago, took weaver ants into the lab. And then on their host plants in the lab, we realized that small black spots were coming up on the leaves. And it's especially visible under lab conditions because there's no rain or dew to dissolve the spots. So you see them more easily in the lab. And then we were trying to figure out what causes these black spots. And we realized, just to show you, they can be very pronounced under lab conditions, as we see here. So what is causing these black spots? And we found out that these are urine spots produced by the weaver ants. And urine is something that animals produce as a waste product when they digest protein. So urine contains a lot of nitrogen, which is a fertilizer for plants. So we were trying to figure out what is the chemical composition of ant urine. And we found that nitrogen was incorporated into the chemical compound called urea. And the nice thing about urea is that urea can be taken up directly by the leaves from trees. It doesn't need to go into the soil and be taken up by the roots. It can be taken up directly by the leaves. And urea is actually something that farmers use on their trees as a foliar fertilizer. If they would like to have a fast fertilization, you can use foliar fertilizers that include the use of urea. So we were wondering whether these nitrogen-rich spots could be taken up directly by the leaves of the plants in the lab. So we did an experiment where we had plants with and without ants, and we also made an arrangement so the ants were prohibited from foraging on the ground. So ants only had access to the canopy of these plants. Then we had plants without ants and plants with ants. And on the ant plants, we then covered some of the leaves so the ants could not deposit spots on those leaves. We were then feeding the ants with an amino acid enriched with the isotope 15N. And then we were trying to see if we can find increased levels of this isotope in the covered leaves. If this is the case, then it means that ants have been depositing spots on the neighboring leaves, and then the spots or the nitrogen has been taken up by these leaves and transported within the plant to the covered leaves. And this is exactly what we found. Here we have the content of 15N, and you see a significant increase in the covered leaves compared to leaves where there has been no ants. So we actually know that nitrogen are being taken up by plant leaves. And you may imagine that if this nitrogen comes from pest insects that have been eaten by the ants, then the tree will realize that a pest insects are being turned into a fertilizer by the weaver ants. What we don't know yet is how big a contribution is this. Of course, I don't think the weaver ants can supply the trees with everything they need, but at least they give a small input to that part of a plantation's economics. Yeah, so that was fertilization of plants. I mean, the last benefit to be derived from ants is protein production. As you may be aware, when ants feed on protein, including insects that they knock down from the trees, then these insects are transported to the ant nests and they are used to feed the ant larvae. So the more protein that is available for the ants, the more ant larvae will be produced in those colonies. And you also know, especially people from Thailand, that these ant larvae are harvested big time right at the moment. Right now, I guess, is the season, because now the weaver ants are producing their queen larvae, which are the big ones that are the most valuable ones. These are being produced up until Songkran, the beginning of the rain season. So I guess people in Thailand are busy harvesting these ant larvae at the moment. So this could, of course, provide a second income to your plantation if you are farming weaver ants in that plantation. The question is whether you can combine the harvest of ant larvae with the use of ants in biocontrol. I mean, if you harvest too many larvae, maybe you destroy the ant population and then you have no control of pests in your trees. So Dr. Vivatbitaya and I made a study some years ago. Desha Vivatbitaya is from Kasetsart University. And we did a study where we tried to find out, can you harvest ant larvae without destroying the ant population? And this is the main result from that study. Here we have an index showing how many worker ants are on the trees over time. And then we have two different treatments. We compare trees where we harvest ant larvae with trees where we don't harvest ant larvae. And then we had one ant harvest in late February, and we had a second ant harvest in that plot in early April. And if you follow the two lines, you will realize in the short term, there's no significant difference between the two treatments. So that means the harvesting of ant larvae did not really affect the worker ant populations in the short run. We also did a measure in October, and now we actually see something becoming close to a significant difference. And then February, the next year, we actually saw a significant difference between treatments. However, you should know that surprisingly, at this point, we find a higher density of workers on those trees where we harvested the larvae. And that might be surprising, that we see more workers when we harvest ant larvae. But there is actually, when you think twice, there's actually a natural explanation on this. And the explanation goes like this: then when the ant harvester harvests the ants, then she will be harvesting queen ant larvae before they have become adults. So they remove ant larvae from a nest, which means that this nest cannot feed these queen larvae to their final size. Under normal conditions, they would have been feeding these larvae until they become adults. But now we have removed these larvae, and therefore this nest have some food in surplus that cannot be used to feed these queens. And they cannot just produce new queen larvae because the production of queen larvae is seasonal. So this surplus of food can only be invested in workers, not in queens. So when we harvest queen larvae from these ant nests, then it will actually lead to more workers because this is the only thing the nest can use their food for. So we actually see that we can combine ant harvests with the use of ants as biocontrol agents. And then we can gain both benefits to the plantation. We also, in the same study, were trying to assess how high yields can you expect from trees in a plantation. So in a mango plantation with mature mango trees, we were feeding our colonies once a week during a 4-month period. And as you saw, we were harvesting ant larvae 2 times. And this led to between 250 to 400 grams of ant larvae per tree. So these are the amounts that you could expect under those circumstances. And as far as I know, this is the only study trying to assess ant larvae production. But I would guess that these numbers could easily be increased. For example, if you feed for longer periods and feed maybe more than once a week, and if you harvest not only 2 times, but more times during the season, then you may be able to double these yields, I would expect. So these are the production numbers that you may expect in this case in a mango plantation with mature mango trees. There has also been done a study trying to calculate the return rates. How much will you get in return if you spend money on feeding your ants? And in this case, you may consider protein either as a free source or as a commercial source that you need to pay for. But if you do these calculations where you assume that you need to buy the sugar for the ants, but protein is for free, it could be insects that are naturally abundant in the trees, or you could feed them with kitchen leftovers. I mean, weaver ants will accept almost all kinds of different proteins from kitchen leftovers. So if you only need to buy the sugar and have free protein, then for each dollar you spend on food, you will have a production of ant larvae worth $4. So the return rate is 4. You could also buy both products. You need to buy the sugar, and in this case, you can also buy the protein in the form of dog pellets purchased from a supermarket. And in that case, it still pays off to feed the ants with a return rate of 1.05. So it actually seems that it should be possible to make money on feeding your ants. And that, of course, will give you a higher protein production, but it will also give you more dense colonies and increasing the control against pests. And what I think is the neat thing about weaver ants in this case is that we actually see that if ant larvae production is based on ants feeding on pest insects, then we actually see that a problem becomes an income, because weaver ants are turning pests into edible protein. So you may argue that a waste product or a problem is being turned into a solution. Weaver ants turn waste into gold. And this is what I think is so neat with the use of weaver ants in tropical plantations. So just to sum up the benefits, weaver ants lead to pest control and to fertilization of the plants, which leads to more fruits. And I mean, if you switch from using chemical fertilizers, then you may actually switch to organic production and get a surplus, a premium price on your product, which is also a benefit, of course. But then also ants produce edible protein in a highly sustainable way, which gives the farmers an extra income. So these are the multiple benefits by weaver ants. And then I would like to mention one more benefit. It's the same story, but putting it in another way, because you may also argue that farmers can save land. And I mean, this is what we need in a world with a growing population and with no more land available for agriculture. Then if we can save land, we are in a good position. And when you use weaver ants, one plot of land can deliver at the same time both a plant and an animal production. So to try to illustrate it, if you are using the beaver ant technology, you have trees with weaver ants. So ants and trees are using the same area. And then you have a fruit production, for example, mango, and ant larvae production on the same piece of land. The alternative in conventional production would be one piece of land with trees to produce the fruits, and then another piece of land to produce food for your livestock. Could, for example, be cornfields that are used to produce chicken meat. But if you use the weaver ant system, you have both of these productions within the same area. So you are actually saving a lot of land on this. Okay, I'm almost done. I would just like to bridge to the next presentation and show you one single more benefit that can be the result of the use of weaver ants, which is carbon sequestration. I mean, if weaver ants can provide these incomes, then of course trees might be more attractive than they would be otherwise. And this could lead to the fact that farmers will plant more trees to use the weaver ant technology to increase and stabilize their incomes. And that means if they start to plant new trees, then we will also have carbon dioxide sequestered in those trees. And this carbon dioxide sequestration can be sold as carbon offsets on international markets, providing yet one more income for the farmers. And this is what Green Ant is about, and which Maria will tell you about in the next talk. Because Green Ant will help farmers commercialize their sequestered carbon dioxide if they plant new trees, making these carbon offsets available on international markets and offering a payment to the farmers for this service also. Of course, it makes sense that the tree growers are the ones that benefit from this carbon dioxide sequestration. But Maria will get into details on this in the next talk. And then just to sum up again, ants can provide more fruits via pest control, fertilization, and they can provide extra income by protein production. And now we have one more benefit. They can also provide an extra income if farmers plant more trees to sequester carbon dioxide. So with these words, I hope I have convinced you about the benefits of using weaver ants in tropical plantations. And with these words, thank you very much for your attention.
Thank you very much, Dr. Joachim. So it is very interesting. So the topic is on the weaver ants, and this is definitely moving toward the SDG economy, the BCG economy, and the work toward the SDG achievement. So that's interesting on producing more fruits, and also could be the pest control. That's great for the tropical zone. I even see a lot of weaver ants at my house in the mango tree as well. So now I will tell my family, don't kill the ants.
Don't kill them. Yeah.
Yeah, so it can be the pest control. All right, so now go to the next session on the green ant. Thank you very much, Dr. Joachim. Continue. Dr. Joachim will be Mr. Mario Simarco. He is a founder and CEO of Green Ant Company from the Netherlands. Mr. Mario has run the podcast Utopia since he was 10 years old. Wow. He is currently an international student at Leiden University College. He is based in The Hague, in the Netherlands. So I would like to welcome Mr. Mario to share about business opportunity with ant farming. I think he will touch on green ants and the ecosystem of raising ant. Please, Mr. Mario.
Thank you very much to everyone, to the organizers, to TISTA for organizing this event today. I agree with Dr. Joachim that webinars are a way of gathering skills and insights from around the world. And therefore, I would like to thank you for all the organizers for this great opportunity, and also the speaker that preceded me, Dr. Joachim. We founded together Green Ant, and he has been with us working in Thailand last month, and we launched training courses for local farmers in cooperation with TISTA, to who we are very thankful for this. So I will give you an insight of what we are doing at Green Ant. I will maybe just share my screen. Yeah, so Green Ant is all about ants and blockchain. The name comes from the queen ant, which is green, which is bigger than green. And for us, the ants are the main starting point. As you can see, our logo is a pink ant, because we believe that the ants are one of the best ways for transforming a cost into a revenue, and are one of the best ways for making agriculture more sustainable and promoting reforestation. So as Dr. Joe explained, we need to increase ant farming if we want to achieve a more sustainable agriculture around the world. But Green Ant is not all about this. Green Ant starts from this. Green Ant supports ant farming around the world because wants to sequester carbon, and using carbon sequestration as a way of including farmers into decentralized finance through blockchain. So we want to build a system in which farming ants is the pillar of an innovating system of farming, which is much more sustainable than before. We aim to build that shift that is needed to contrast effectively climate change. Climate change touches every level of our lives. And if we want to contrast it, we need a radical shift at all the levels. So how can we make organic agriculture and carbon sequestration blooming in Thailand, giving everyone the resources for implementing this without losing? It can be that planting organically, farming organically, is a privilege of few that can afford it. It can be that investing in contrasting climate change is the privilege of few people. It should be profitable investing in these things. That's why we are based on ants, because with ants, it's profitable investing in organic agriculture because the ants are extremely efficient. But this is not enough. We need to have a system that offers alternative. For example, the ants offer an alternative to unsustainable pesticide and are an alternative protein. But we need also, if we want to really achieve something in carbon sequestration to be able to monitor the amount of carbon that each tree sequesters and reward people who are sequestrating trees. The carbon offset market is extremely flawed around the world because it doesn't really reward those who every day take care of growing the trees and making them organic. That's a failure of the rewarding system. But with blockchain, we can go ahead of it, monitoring better, offering alternatives, and rewarding better. So what we use? Well, we use Weaver Ants, and I'm not going to explain more about the Weaver Ants, but we also use decentralized finance. We use blockchain for giving the resources to the farmers to implement organic farming and plant more and better trees to sequester carbon around Thailand and around Southeast Asia. And we use artificial intelligence to monitor the exact amount of carbon that each tree stores in order to have accurate data. So what are the benefits of decentralized finance? Decentralized finance, being based on blockchain, doesn't need huge institutions to be trustworthy. This means that it can include people even in conditions in which financial institutions are not really accessible. And therefore, that decentralized finance can be extremely inclusive, much more than traditional finance. And decentralized finance, being inclusive, can give access to capital to farmers that then can invest in independently planting. And this is throughout the world a problem. We started from Italy, where we found the problem that raising capital for the farmers was a huge problem because they had to borrow money from people who were also selling them pesticide. And from that, we started believing it that an alternative finance was needed if we wanted to stop the dependency from pesticide and borrow with capital. Through decentralized finance, we are able to implement new technologies and rewarding farmers for farming sustainably. But we are also able, through blockchain, to trace the origin of the products that we're farming, rewarding even further farmers who are farming organically, allowing them to sell on international markets. For example, through our blockchain and our AI monitoring, we will be able to monitor the presence and gather data of the trees which have the ants to certify that those trees are farmed without pesticide. Because as Dr. Joachim explained, ants do not survive in presence of pesticide. So their presence certifies the non-usage of pesticide. And all of this stimulates sustainable carbon sequestration and reforestation. But how do we do this in practice? So in practical terms, we take a tree, we take the GPS coordinates of the tree, we take the species of the tree, and we create an NFT. An NFT is a non-fungible token. It is a blockchain product that has the main characteristic of being immutable and unique. So we create an NFT for each tree planted and tokenized. Each NFT generates one green ant coin for each kilo of carbon stored in that tree. And therefore, the farmers find themselves with two plantations. One is the real plantation, and the other one is the virtual plantation. One plantation produces fruit, one plantation produces timbers, the other plantation produces green ant crypto coins, which are convertible and spendable can be exchanged to fiat money. So what we are building is a system through which the farmers can take their trees and access with them decentralized finance, attracting investment towards tree planting and carbon sequestration. So we build our network around sharing and exchanging NFTs in order to be able to increase the amount of carbon stored and the amount of the verification of our data. Data verification comes to be a main pillar of what we're doing, because if we are basing a cryptocurrency on the amount of carbon that we are storing, we cannot go wrong on data. Data are the very basis of our product, because the exact amount of carbon stored generates the crypto coins. But the data are also able to guide us toward a better reforestation projects and guide us throughout scientific publications, scientific researches. So we are right now cooperating in Thailand with Tister and with many farmers around the country, and we have seen a remarkable interest in farmers to start farming with their hands and to tokenize their trees for accessing decentralized finance. And this is very interesting that there is this interest, because when we started, we didn't expect the farmers to be so much interested in accessing decentralized finance, or that accessing decentralized finance was such a need around the country. And instead it is because raising capital and be able to cooperate in the fight against climate change is a widespread interest. And therefore we should join scientific institution, organization, we should join all together to promote this on a larger scale. Because this represents a very large shift in the way in which farmers farm the land and finance and manage their own economy. And therefore, for being able to achieve this shift in the best ways, we need to cooperate all together and to manage to have the best strategy. I think that this was more or less what the main part of my presentation. But it will be very nice to have a debate with the people present here to reply to any questions. I'm sure that there will be the opportunity to do this, because maybe people are curious about what Dr. Joachim, I and Tister and the entire Green company is doing for promoting this shift. So thank you.
Thank you, Mr. Mario. So it is an interesting network that you have built, Green Ant. Right now, we are going to have the last speaker, the winner of the Vital Innovation Award. And Mr. Mario, Mr. Joachim, we will invite you back for the question and answer later on after Ms. Sandra Barcel will be finishing her presentation. Our next speaker from the speaker from Vital Innovation Award 2021 Smart in Food, insect-based food source to supplement nutrient deficiencies in vulnerable areas we have with us here Dr. Sandra Balcel. She is the R&D Senior Researcher, Bioresource and Agri-Food Technologies, Environmental and Biotechnology from LEITAT Technological Center from Spain. So she is with us here. I have a bit of a brief of Dr. Sandra. She has a degree in biology in 1998 from University of Barcelona. She also studied industrial biotechnology and proteomics, food safety management, and insect as food and feed from Wageningen University. And interestingly, she is the winner of Vitro Innovation Award 2021, currently working in Circular Economy Department of LEITAT Technological Center as Senior Researcher in the Bioresource Agri-Food Group in the execution of research projects and preparation of several national and European proposals. Throughout her scientific career, she has participated in many different research lines, including byproduct valorization by applying physical, chemical, and enzymatic treatments for the recovery of proteins and production of protein hydrolysates and bioactive compounds such peptide, fatty acids, polyphenol for food, feed, and agriculture application. So recently, she focused her research on insect processing for the recovery of protein, lipids, and chitin. So right now she is with me here. May I invite Ms. Sandra Balcel to cover on her talk on smart in-food insect-based food source. Please, Sandra.
Hello, thank you very much. Yeah, show my screen.
Yes, please, share screen.
Okay. No, okay.
It's all right now. Yeah.
It's all right. Okay, okay, good, thank you. Okay, thank you very much for inviting me to this webinar. And special thanks to organizers. I'm going to present the Smart in Food project, which is the insect-based food sources to supplement nutrition deficiencies in vulnerable areas. This project was awarded by Vitro under the framework of future equity and sustainable agriculture, to contribute to the UN Sustainable Development Goal, Zero Hunger. Firstly, I would like to introduce LEITAT. LEITAT is a technological center in Spain. It's a private non-profit organization that offers solutions to the technological needs of companies and entities. We are based in Spain, and most of our working areas are in the north of Spain, in Catalonia, and we have also a working area in Chile. We are around 400 people, and we develop different projects, national projects, European projects, and also international projects and services. We have 5 areas, basically, yeah, health and biomedicine, circular economy, energy and engineering, applied chemistry and materials, and also we offer advanced technological services. These areas are closely interconnected. And our project is based in the circular economy. Our department, Bioresources and Agri-Food Technologies, belongs to the business unit of circular economy. And here we develop different projects. We valorize different waste streams and bioresources. For example, animal waste streams, byproducts, and also vegetable byproducts, agriculture, basically. And also new alternative sources like microalgae and insects. We develop different processes to extract different ingredients. And then finally, we formulate these ingredients to food, feed, or agriculture, into fertilizers or biostimulants. To develop these processes, here we apply different technologies, basically to extract protein, and also other bioactive compounds like polyphenols, peptides, and for example, other compounds that can provide different functionalities, for example, antioxidants. And regarding the final application, here we can apply different technologies to adapt this ingredient to the final formulation, including here, for example, the food 3D. So regarding the insect technologies, here at Lated, we cover the full value chain from production to the product development. Regarding the production, we start these tasks by identifying the different waste streams that are available at local level in significant amounts. Then we analyze these waste streams from the nutrition point of view, and also we analyze the contaminants, the chemical contaminants, and microbiology. Then we select the optimal waste streams, and we formulate these waste streams into insect diets. Then here we optimize the process for insect breeding. And once we collect the biomass, here we can apply different technologies depending on the final goal of the project, but basically drying, fractionation, functionalization, and texturizing. And finally, we formulate these ingredients into the final product, food, feed, or also other applications. For example, we can work here with chitin and develop products as fertilizers, as biostimulants. One of the examples is the project of Susin Chain. This is a European project, and here we participate in food and in feed. In feed, we process the larvae of Aromatia. We apply enzymes, and then we produce the insect meal. And in food, we formulate different insects meals and into different products, for example, sausages. So the smart in food, the problem. As you know, the food demand is expected to increase by 70% by 2050, over 9 billion population. To this, it is added the nutritional deficiencies, which can be more severe depending on the geographical area, economic development, and climate change. Worldwide, 0.8 billion population suffer with chronic hunger and 2 billion with nutritional deficiencies. In South Africa, anemia prevalence is around 17% among adults, and there are target groups that are vulnerable because of they don't have balanced diets. They are at risk because they are, for example, children, elderly, or people who suffer from chronic disease. There are groups that are vulnerable in different areas. So the food system is facing the perfect storm. On the one hand, there is this increase in the population and consequently in the food demand. But yet 33% of food is wasted. And on the other hand, we have this the conventional producing food systems that produces a lot of greenhouse gases and consumes lots of resources, water and energy. So to meet the goals for 2030, we need to reduce food waste and CO2 and reduce hunger. And what's the solution? Alternative proteins can be part of the solution. And in fact, in the market, there are companies that they are already working with different alternative proteins, for example, plant-based proteins, proteins for fermentation, algae proteins, insect proteins, and culture meat. The focus today is insect proteins. And you can see here some companies that are working with insects and the formulation of insect-based food products. And as you can see here, the global trend is growing exponentially. The market size for proteins are increasing constantly. And what's relevant in alternative proteins? Well, basically, there are three main pillars: the nutritional profile and organoleptic properties, the micronutrients to cover deficiencies. This is the key of the project in Smart Food, and the sustainable and animal welfare. If we look at the protein, insects are a promising source of protein. The content it changes depending on the insect species. And here you have on the right a comparison between beef and grasshopper. As you can see, insects are a good source of proteins. The content in grasshopper are quite similar to beef, while fats are lower, and calcium and iron are higher. And this is very important because this is not only protein, this is also that minerals and micronutrients that can be deficient in some areas. And for example, in the case of iron, what's important here is also the availability, because the uptake of this iron has been reported to be, in some cases, even higher than in beef, for example, buffalo worm in this study. So insects are a good source of not only protein, but also these micronutrients, among them these minerals, and also vitamins and other compounds that are crucial and essential in some regions to formulate diets. And the last pillar is the sustainability and animal welfare. Insects are a good source of protein when they are converted in insect biomass. And what's important here is that they can grow on organic waste, agriculture byproducts, waste streams, so they can reduce this waste and they convert this feed, this substrate, into protein, and to do so they are highly efficient. For example, to produce 1 kilo of protein, the insects use 1.7 kilo feed, while cattle need 20 kilo feed. That's a huge difference. Another advantage is the water. Insects use less water, only 3% of that used in cattle. And another advantage is the land. They use less land. They need a smaller space, and in addition, they produce less greenhouse gases. So this food system it's a promising food system to provide proteins and also micronutrients, and because of these advantages. The solution in a Smart Food project. The core of the project is the circular economy, as I commented here. And what's important here is that we are insects on local agri-food byproducts. And to do so, what's important is that here we identify the waste stream that are locally available in significant amounts. And to do so, we will contact different providers along the food value chain, including fresh food markets, distributors, and so on. And this will be a base solution for that specific area, including the processing of these waste streams to prepare the insect diets. Then insect weaving will be optimized and processed into different proteins, chitin, and oils, and finally formulated into feed and food. Here you can see that we develop fast food. And why fast food? Well, as I said, among the target groups, there are children that are vulnerable to the nutritional deficiencies. And the goal is to make insect-based food products appealing to the target population. And to do so, we also include here the packaging, nice packaging to make this attractive. So there will be 3 case studies in this project, 2 of them in Africa. In Africa, agri-food byproducts will be identified and selected to rear specifically mealworms. That's the Tenebria monitor. Once rear, the insect biomass will be processed, and the parameter here will be optimized to enrich the proteins and minerals, specifically iron and zinc. And finally, as food products, snacks and drinks and burgers will be developed, including also nice packaging to reach the population. And similarly, in Thailand, here the goal will be byproducts from bioethanol biorefinery. In this case, these byproducts will be formulated into diets for crickets, and finally processed into insect meals, here in this case to develop feed for poultry and fish feed. Here the goal is to provide alternative proteins for feed, and not only as a protein as itself, but also as a supplement to provide some health benefits, because insects can provide benefits such as antimicrobials, and also can boost the immune system of the animals. As a value proposition, I would like to highlight here the fact that this will be a local-based solution, because local byproducts will be specifically identified and selected based on the availability and characteristics, and specifically composition. It's important also the involvement of the citizens, because they will be involved in this project from the early beginning of the project, because we need to select these food value chains. And as benefit, we expect to increase the food availability in these target groups, and also the protein availability in feed. The partnership now includes 4 technological centers, LEITAT in Spain, then FIRO in Nigeria, CSIR in South Africa, and TISTR in Thailand. The 2 case studies in Africa will be focused on the rearing of mealworms to develop food products, and the case study in Thailand will focus on crickets to produce insect meals to formulate feed. So the roadmap of the idea of the project, of the Smart Input project, includes the first phase, that is the Data Innovation Award. The second phase, that is the project, the R&D project development, and finally the phase 3, that is the industrialization. In this first period, that is the Data Innovation Award, milestones are the project positioning and dissemination, the creation of a stakeholders network, and initial studies and waste identification and characterization. So the idea is to position the project And to do so, we need to involve other companies. As I said, we are now 4 technological centers, research centers, and then we will incorporate to the consortium end users, food companies, feed companies, also partners with expertise in terms of exploitation. In phase 2, this will be we need to identify a call and then prepare this R&D project to cover these milestones, that it's the insect diets formulation, the development of food and feed products, the validation of these 3 case studies, and then the final goal is to develop guidelines for insect breeding and processing at local level, but that we can adapt to other regions by modifying the formulations and the substrates. And finally, phase 3 is that industrialization. Here, the main goal is to replicate these case studies in other regions. And last but not least, the market size. Let's have a look at this slide. Here you can see the global market for proteins at protein demand market, protein market size and animal feed at global level. And on the right, you can see the market size for proteins in the participating countries: Nigeria, South Africa, and Thailand. What is clear is that protein global demand is increasing exponentially. Protein global market size is consequently increasing, and also the feed global market size. So the Smart in Food project offers a promising solution to provide more protein and food, and at the same time cover these nutritional deficiencies, because the best way to predict your future is to create it. Thank you very much for your attention.
Thank you very much. Thank you very much, Sandra. So I hope that will be a lot of ongoing in Thailand. I see now working with tester more on the cricket and on the feed. We also see a lot of development in terms of going for food as well. We hope we can do more and more together with your center and also the Green Ants. Right now, come to the Q&A session. I would like to invite Mr. Mario, Dr. Joachim to join on the question session. All right, so let me ask one of the questions from the audience here. To Dr. Joe, the question is, I wonder if the nearby farms use the strong chemical pesticide. I think we have the land, the neighbor use the strong chemical pesticide. Will ants in our land still efficient to control the pests?
Yeah, I would like to say that first of all, weaver ants are very sensitive to insecticides. So if they are exposed to insecticides, they will suffer from that. But the distance where you see drift from one neighbor to another neighbor, I think drift takes place only for short distances. For example, we did a project where we compared trees that were sprayed with other trees that were protected by weaver ants, and we were just putting up some screens, and one line of buffer trees between the 2 treatments was, and then some screening. So I think the effect from a neighbor using strong chemicals would be limited, but there will be an effect on those trees that are closest to the spraying.
All right. So come to the next question about the protein, like the profile of the nutrient profile of the protein that come from ants. Have you had any experience on analysis of the profile of the proteins that extract from weaver ants? Any idea to share here?
I am not aware of studies that have So do you mean what kind of proteins?
Yeah, the nutrient profile, because to use it, the protein from ants to in the either feeds or either food, the food scientists must see the nutrient profile.
Yeah.
This question when I moderate one of the session on some insect based, the question from the user always asks, what is the nutrient profile that we can really bring in from the ant proteins? Is that up to that step yet, or it's on process?
No, we have measures on this, and I mean, but it's very different from the different stages. For example, a larvae, just before it hatches into a pupae, the larvae will be very high in fat. They also high in protein, but they have a high fat content just before pupation. Then during pupation, they will burn out that fat, and then they will be very lean as a newly hatched pupae. And I know in Thailand, for example, you hatch both the larvae, the pupae, and also the very young adults. So depending on what stage that you feed on, there will be different nutrient profiles. But some of these stages have very high protein content, like 50% on dry weight basis. So they are comparable to most other insects.
Yeah, all right. So it means that there is some analysis and data keeping in each stage. So that is available for research and do the further study.
Yeah, it's important if people would like to study this, that they separate the different stages, because they are very different.
All right.
And those studies I have seen so far they have not really explained what stages did they use in their studies.
Okay.
So that's an important point.
Very good like pinpoint on the stage of the ants. That's very important. So I will go next to Mr. Mario. Mr. Mario, you have mentioned about blockchain, about the green ants. Can you explain a bit more if anyone want to join in the program of green ants, how they should be in contact, or what is the facility they need to have in order be part of doing this research?
Yes, thank you. Thank you for this question. To join our projects, you need to be committed in farming trees for at least 10 years. And this is because less than 10 years, there will be no environmental impact over carbon sequestration. So our main requirement is farming trees for at least 10 years, but also farming them in an organic way. So we suggest to use weaver ants as a way of being sustainable, as a way of producing protein, and using an alternative sustainable pesticide. However, we also promote organic farming holistically, and therefore the sustainability in agriculture. So joining us is possible through our website. You can join, and reaching out to us, making your for now you have to write an email and ask if you want to join, and then we will tokenize the trees and start monitoring them.
Mr. Mario, to extend to that question, seems like anyone that is interesting would be able to join the program. I take assumption that most of that interest would be the one who already have their mangoes field, have their farm, their plants already. So that's any specific requirement should be any type of plants that can do, or any prioritize of the species of the plant that can join the project, or every type of the trees, the plants.
So our aim is being able to tokenize as many tree kinds as possible. However, for the moment, our AI is not able to monitor every kind of trees. So we have to focus on certain kinds of trees only. And therefore we focus on mango and citrus. But anyone is welcome to apply, and on the long term, we will welcome anyone who is able to provide evidence that they're farming organically and that they are committed in farming those trees for at least 10 years.
All right. So right now, only two. Any factors affect apart from the tree itself? It has to be in the area that has more rainy seasons and any other specific, yeah.
The verification, the monitoring of the trees is cyclical. So we usually work on a 6 months by 6 month basis, but on the basis of the specific condition of the piece of land, this will be arranged with the farmer. So there are no specific requirements in those terms.
So I come to the next question. Maybe Dr. Joachim and Mr. Mario can help each other to share your idea. The farmers in Thailand, most of the business is from father, mother, from ancestor doing the farm for a long time. Speaking English, I would not say it's easy for them, and to change their mindset to do something about growing ants. The ecosystem sustainability is kind of it just stepped in like 3-5 years ago, and pressure just come on now. How you gonna convince Thai farmers in terms of adopting the projects? Any idea from Mr. Mario and Dr. Joachim?
Well, maybe I don't know if Joachim wants to go ahead, but just for summarize, I believe that the environments, the economical environments in Thailand is extremely favorable for doing this. Because the main pillar is that because land is divided among small landowners, and for a matter of tradition is farmed, people really believe in the value of their land. People really take care with their hearts of their land. And therefore this helped us incredibly in dialoguing with the farmers about the need and the necessity of building this, because everyone cares by every trees they have on their land. At Green Ant, we monitor each tree. We take care of each pieces of land. So we believe that instead the system of holding the land is actually very, very suitable, much better, much more suitable than in other cases in which land is owned by large corporations, who don't really care about every rai of land. And for the translation, for the English, well, I don't speak Thai, but instead we found that the younger generations, the younger farmers speak English and have a basic understanding of blockchain technology. And overall, people of all the ages have wide interest in innovating through Weaver ants and blockchain. So actually, yeah, it's surprisingly, Thailand has been surprisingly innovative when it comes to be taking care of the land.
Yes, agree. Thank you so much. Dr. Joachim, any things to add?
Yeah, I would just like to put a small note on that. I think actually Thai farmers are very adaptive. So, and again, I mean, the use of weaver ants is very straightforward in Thailand compared to many other countries, I think, because the farmers know that these ant larvae is valuable. I mean, they see people harvesting these ants, and I think actually the prices on weaver ant larvae are increasing. we visited one place in Thailand where they had a can. So now they have started canning the ants. And I think the kilo price of ant larvae that were canned were like 2,000 baht per kilo or something. So it's very easy for the farmer to see this very clear benefit of the ants. And also I think when they start seeing somebody have success with this, they will be able to adapt. I think I find Thai farmers rather adaptive.
Yes, definitely it is. I think it's because we already have the weaver ant soup, you know. So
yeah, exactly.
A lot of weaver ant soup, that is a kind of benefit, and we hope that with the with the communication campaigns to promote, I think a lot of farms in around Thailand, they can adopt and understand that. Talking about blockchain, that is not that difficult, you know. When the wording blockchain, AI, they kind of can bring the fear, but I think they love the land, as Mario said. Every tree is their value. It's really good opportunity to sustain their farm and also create value to the farm. I really like the idea and feel like we need to promote more of this and extend from mango itself. All right, so now come to Sandra. Sandra, I did a lot of moderating session with black soil fly larvae, crickets. Everyone is talking about feed and food. And now it's good to know that cricket profile or the approval from Europe, Australia already there. How you see the value of the adoption? I saw that the focus more in feed. How you see the food segment in terms of using the insect beds, improving or growing? Sandra.
Yes, I think you are asking me about food and feed, Seitan. Yes, in both sectors, because we need new proteins also to formulate feed, because the demand for proteins in feed is increasing as well, because as the population grows and also some middle class, they ask more for meat proteins. So we need alternative proteins in feed, also to reduce this volatility in prices, because we need to, all countries need to import feed, and so proteins specifically proteins from feed like soya, yes, and then we need alternative proteins for feed, and also for food, of course, because the population is growing very fast, and so we need to combine these. Insects are a good source of proteins, and also the other alternative proteins can provide extra proteins for food.
Yeah. Sandra, it seems like the problem now is like the price, the cost of the proteins from insect comparing to the current source of protein. How, as from the researcher perspective, how you see the development from country to country or region to region? How do you see this? Do you see the trend of the price coming down in how many years, or how you foresee, because you interact with a lot of research that has been done?
Yes, you are right. At the moment, insect proteins are more expensive, and sometimes, the price is difficult to compete against the conventional proteins. But this is because insect farms are not very large. But this is a technical issue that it's evolving very fast. Now there are bigger insect farms, and they are and the improvement and the technology is practicing quite fast. They are automizing the different real faces of the insects, and the fact is that once the insect meals are produced in bigger volumes, the prices will be reduced, or at least it will be easier to compete with other alternative with the conventional proteins. I mean, but another issue is the quality, because protein profile and insect biomass is healthy. As I said, that the fats are lower, the minerals, they are a good source of minerals, they can provide extra benefits. There is not, I mean, there are different, if you compare with cattle, for example, they need in the conventional farm, they need antibiotics and other external substances that are added to the feed. And in this case, the advantages is that they are healthy in this issue, in this aspect, and the profile, because it's a good source of proteins and minerals.
Yeah. So I saw from your slide, you have like one stage called waste volatilization. So it seems like the agro waste in Asia, I would say 40% agriculture in go to the waste after post-harvest. Have you had any plan or any project that is going on in ASEAN countries in terms of utilization of this type of insect-based project to support the reduction of the waste from agriculture?
Yes, well, we have TIS. They are involved in the project, and they have experience with crickets and the reuse of waste streams from the bioethanol industry. And they will participate in this project, and they will provide this experience. And yes, the idea is to make the insect farming feasible at the local level, and while respecting the traditions, the culture, the citizens, the processing systems. But yeah, the thing is to select also the technologies, because we need to implement technologies that are feasible to that region. That's the novelty, the idea to adapt the process to local level. And now we have currently some ongoing projects, and later we have national project and also European projects, like this project that I commented, Sushin Chain. And yeah, the idea is to develop international projects as well, because this will provide more benefits to the whole consortium and the companies that will be involved in the project.
Yeah, very nice to hear that, because I also work on agro waste. That is a huge agriculture waste in our region. So now I would like to back to Dr. Joachim, and maybe Mario, you can add, since I want to ask about green and projects. So I want to know if I have the farms already, but I'm wondering that to invest in the technology together with green ants, how is the investment going to cost me if I am the farmer who is interested to work in the project? Is it a big investment or so or?
Very small.
No, I don't know if Joachim wants to say something.
No, I think it's your question.
Well, it's really not a big investment. I mean, farming ants is not requiring anything more expensive than basic equipment. and generating NFTs is free for the farmers to join. So it is all about gaining. The risk is very, very minimized.
Yeah, but you mentioned about 10 years. So they just invest for the beginning, but the result to catch from the ecosystem, it needs at least 10 years of commitment. Am I right on that?
Well, they, in terms of the NFTs and the coins, they get them every 6 months. Like they get the NFTs once and the coins every 6 months, more or less. It depends on when it is monitored. So can vary, but on a general rule is 6 months. And the commitment is 10 years of farming their trees. So obviously there is a revenue coming from those trees anyways, and from the ants farmed to them. But the trees must be being planted for at least 10 years, otherwise there is no environmental impact. So yeah, there is a commitment in terms of length of the contract. But as I was saying, landowners and tree growers in Thailand care about all the long-term benefit. And so for 10 years is something on which we managed to agree with most of the people. It does not be a huge obstacle because everyone wants their trees to be there for a long term. So yeah, that's the only commitment. I don't know if Joachim wants to say something.
Just putting a note on the weaver ant farming aspect. I mean, one of the things that we actually promote about this technology, that it is so extremely low tech. I mean, what you need is a scissor and a piece of rope, more or less, and then you can get going with your weaver ant management in your plantation. The only thing that you actually need is knowledge. And also here we provide free training courses for farmers and also for extension officers if that becomes relevant. So knowledge is the key point, and which is freely available.
And Dr. Jo, to my curiosity, that apart from growing the mango, the government, I'd say Thailand, have good agricultural practice in having the ant growing, the ant farm under that. Do they need to have another ant good agricultural practice, another control, or it's just one good agricultural practice?
Or do you mean if ants can control everything, or they need other measures?
No, I mean that is the certificate that the farms, if you supply the products, you must have GAP, good agricultural practice for your production. But having ants in that facility, do they need to apply another good agricultural practice for the insect growing, or it's just one?
Yeah, I actually don't know what are the conditions for these, but I guess I'm pretty sure that the ants would be compatible with those
mango tree,
with those practices.
Yes. All right.
But I don't know the practices in detail.
Yeah, I just curious because I know that what Sandra mentioned about cricket, we have the GAP implement for the farmers. I would study further for my knowledge as well. Now we have one question from the audience, Dr. Jo, on, do you have any technique to find the ant queen in the mango tree for the ant farm in case we have a lot of ant nests in one tree?
Yeah, that's a very good question. But actually there have been 3 scientific papers made on this, but it's not easy. I mean, finding the queen is key, because if you want to transplant an ant colony from one place to another, you need to find the queen. And there have been some studies. I mean, usually the queen will be in the middle of the colony. One weaver ant colony can cover many trees. So usually she will be in the middle somewhere, and she will usually also be in the top of the highest tree, and she will be hiding usually in a small nest. Maybe you think she will be in the biggest nest, but this is not the case. She will usually hide out in a small nest in the top of the biggest tree in the center of the colony. But also I would like to point out that because of the difficulty of finding the queens, we are also working on establishing ant nurseries. Because during the mating flight of the ants, we can harvest queens by the hundreds in numbers, and then we can nurse them and make them build colonies under controlled conditions, and then we know we can supply a colony, including a queen for the farmers. And
So
the farmers can actually. Sorry.
Yeah, one tree is one colony, but the queen can have many colonies.
Yes, exactly.
And a
Yes.
tree. Yeah.
Yeah.
Okay. Interesting. So I have to find queen of the queen.
Yeah.
So the next question, Dr. Joe, on if in Thailand they harvest the river ants egg from February to May, okay,
Yeah.
what will you recommend if we want to harvest more than one time a year? How to have the whole year?
This is another very good question, because I mean, if you can produce ant larvae outside season, you will also obtain a higher price, I guess. So this would be very important to test. And I have discussed this, for example, with Dr. Vivat Vitaya when I worked with him many years ago. And we think we don't know it yet, but it will be interesting to test whether, I mean, the ants will start producing ant larvae, queen larvae, just before the rainy season. So we have been thinking if we can induce the production of queen ant larvae by spraying ant colonies with water, because we know, for example, from Tanzania in Africa, in that country, in some of the regions, they have 2 rainy seasons per year. And in those cases, we see that the ants produce queen larvae 2 times a year. But if we can trick them with artificial irrigation, I'm not sure, but it's worth to test it, I think, in future studies.
Yeah. Thank you so much, Dr. Joe. If we talking about this 10 years ago, I will say Thailand has 2 seasons, rain and no rain. Nowadays, it's hot and no hot season. So I have to identify whether to balance on commercialization. If off season, definitely the price will be high. And how I would think about like how we can store it and sell it off season for the pricing if we really cannot manage the rain. So thank you so much, Dr. Joe. So now to Mario, the same question, but once again from our audience here. Dr. Joe and Mario together, the audience say it is difficult to convince Thai farmers for your system on ant farming. What was your challenge in convincing the farmer?
This is one for Mario, I think. You have been convincing farmers lately, haven't you?
Because Dr. Joe also spent a lot of time in Thailand. Mr. Mario also focus on the project. So you both can share the challenge.
Yeah. So obviously there are always challenges in convincing and in business in general. But I believe that the support that we have gotten, we have received from Thai communities, Thai farmers, it's been impressive and very unexpected to a certain extent. So yeah, I will say it was challenging. Surely there was a language barrier, a cultural barrier for sure. But I will say it was insignificant compared to the welcoming that we got and the support in general from T-Star, from the communities, from the farmers. So, yeah. So I read here from another question about what's the minimum size of the land. So for joining Green Ant project, there is no minimum size of the land. There is a minimum amount of trees, and the minimum amount is 20 trees. For joining, you need to have at least 20 trees, farmed and grown for at least 10 years from the tokenization point. I don't know if Joachim wants to say something else.
Yeah, I would like to point out that I think the main challenge we met, I was in Thailand some months ago and were lucky to meet some of the farmers that we are negotiating with. And I think the main obstacle we found during that was one of the farmers that were producing very high quality fruits for supermarkets directly in Bangkok. And he was using a lot of chemicals. in order to get these very perfectly mango fruits. And of course, if we tell him that he cannot spray any longer, that will be a challenge for a guy like him. But he actually agreed to try to do a test on some of his trees. But what we met later was an organization of organic growers. So farmers already growing organic. And I mean, these farmers, they will actually have weaver ants more or less. Sooner or later, the weaver ants will show up by themselves. And they have nothing to, I mean, so an organic farmer has nothing to lose at all by joining our project, because they already have the weaver ants. So as soon as they plant new trees, then they will be able to join our program, and there will be nothing for them to lose in that respect.
Yeah, I think there's also pressure by the consumer in the future that everyone looking for the organic fruit. I think that pressure gonna push our farmers to change their way of doing farming.
Yeah, I think so too, that there will be an increasing demand for organic produce. I mean, in Europe, in Denmark, for example, now the market share of organic produce is like 20%, and it's growing very fast from year to year.
Yeah, that's why the GAP story, the good agricultural practice and the traceability of the products is in place there. So thank you very much. So I come to the last question. Today is our honor to have our Retro Innovation Award winner 2021 here, Sandra. I know that Retro doing a lot of work and put together like the researcher organization. I want you to share your experience being as a winner and invite the members here or non-member here to join Retro. Can you share how you feel about it and how you see the benefit of being together in Retro project?
Yes, thank you. Yeah, we are excited to develop this project. And that the fact that the project that was thought at the international level, this offers a great opportunity to share experiences and adapt technologies to the different areas and make the process of insect farming feasible, not only technically but also economically feasible at that region. And the preparation of Retro was a very interesting and exciting opportunity to collaborate with FIRO, CS, IR, and TIS. A great experience, and now happy to start the project. Yeah.
Yeah, Retro also give you the innovation awards and also opportunity to network to all of us. Thank you very much, Sandra, for being with us today. So I will to the last part. Thank you very much, Dr. Joachim, Mr. Mario, Sandra, for sharing today. And we cannot miss it. It is photo session. So I would like to invite the TISTER team and all speakers to from the morning session who join here, Dr. Bandit, Dr. Shen, and Dr. I think many of you here and the team from TISTER to join in the photo session, please. Dr. Kwa, are you here? Dr. Kwa, please. So I promise next time if I have opportunity to host, I would be trying my Chinese speaking. Dr. Kwa, are you here? All right, so we gonna do a photo session now. Let's smile. 1, 2, 3. Another one, please. 1, 2, 3. All right, on behalf of TISTER team and all the supporting organization, I would like to thank you to all speakers and audience in this room. I would like to apologize if I use any wrong word or any pronunciation of your name wrong. Apology for that, and hope the audience here enjoy. The next one on the handouts, you can download from the chat box, and to improve the quality of the webinar by Twister, Whitero, and the other organization that put together this program, we would like to request you to do the evaluation form by scan the QR code here. And thank you very much for joining us today, and have a great rest of the day and stay safe from the COVID. Thank you. Bye bye. สวัสดีค่ะ
Thank you very much.
Thank you very much. Bye bye.
สวัสดีครับ and thank you for a very good
สวัสดีค่ะ
moderation.